Good morning, everybody. Welcome to this multi-stakeholder workshop on the data quality framework for adverse drug reaction reporting. Thank you all for joining. I'm just going to do a little bit of housekeeping before we kick off with the workshop as a whole. So participants should note the following. The meeting's broadcast and recorded. By attending this meeting, participants are consenting to recording and broadcasting. By default, everyone's muted to minimize the risk of potential disruptions. If you wish to speak or make a comment or ask a question, please either await the Q&A, and there's a Q&A session at the end of each little block of presentations, or there's discussion sessions towards the end of the day as well. You can raise your hand by clicking on the icon in the lower right corner of your screen, and when the chair gives you the floor, you'll be unmuted by the host. Once you've spoken, please click on the icon again to take your hand down. But you can also post your questions in the chat box, which you'll see on your screen as well. The chairs will give the floor in a way that tries to balance stakeholders and respects the time constraints. Obviously, this is a constrained meeting and we'll be finishing about 4 o'clock. We may not be able to address all the questions during the workshop, but if your question hasn't been addressed or won't be, it looks like we won't be getting time, please do use the chat box and be assured that we will read all of them and take good note of them. Slido is open to the audience. That will be used during the final session, and it also includes those of you following on the live broadcast. Instructions on how to access Slido will be visible on the screen before we launch any Slido polls. Now, that's the housekeeping over. I'll hand over to Georgi Genov, the head of pharmacovigilance, for the welcome and introduction. Thank you.
Thank you very much, Tom. Good morning, all. It is my great pleasure to open this multi-stakeholder forum on the data quality framework for adverse drug reaction reporting. I would like to thank you all for your participation and express my appreciation for your continuous collaboration on our journey to data-driven medicines regulation. Pharmacovigilance is evolving faster than ever before, making use of greater range of data sources for signal generation and validation, adding to individual databases the world of big data. In order to characterize, compare and combine the various data sources and the outputs which will lead into decision making, it is necessary to have an overarching framework of data quality. This is what the heads of medicine agencies and the Big Data Steering Group tasked EMA with creating and what was delivered last year. This framework will enable the expansion of data sources to strengthen data analysis across the community so that potential safety concerns can be rapidly and robustly reviewed, and actions taken to protect public and also animal health. By delivering the big data vision, we can also get medicines to patients more quickly and optimize their use on the market. So, turning to today, we want to take stock of achievements made, but also look ahead. We'll bring you all up to date with the current published data quality framework. We will take deep dives into the data quality activities currently performed by the EA regulators, both human and veterinary. We will also have presentations from human and veterinary marketing authorization holders, a CRO and a non-commercial sponsor regarding the data quality activities they perform and the challenges of obtaining and providing good quality data on ADRs. And we are privileged to have an expert panel of great experience to discuss the challenges related to recording, measuring, characterizing and analyzing ADR data quality. Lastly, we'll have an open session for you to share your views on the challenges you face in obtaining and reporting on ADRs and to discuss what topics you think should be in the final chapter of the framework document. I encourage you all to use this meeting as an opportunity to provide your views and share reflections on your ADR data quality work. I would like to thank you all again for engaging in this very important event, and I wish you all a very productive day. Thank you. With that, I'll pass to Paolo.
Good morning, everybody. I'm Paolo Alcini, Head of Healthcare Data here at the agency in the Task Force for Data Analytics and Methods. I would like to introduce Anja van Aren. Anja has been working in the Pharmacovigilance Department of the Dutch Medicine Evaluation Board since 2008 and has over 20 years of involvement in eudravigilance-related work. Anja is co-chair of the Eudravigilance Working Group and the EMEA NCA Pharmacovigilance Business Team. Also, she is a EU representative in the ICH E2BR3 and ICH E2D R1 expert working group. Anja, over to you for the opening remarks.
Yeah, thank you, Paolo, for introducing me and for having me in the workshop. I'm trying to share the slides. Can you see them?
Yes, we can.
Okay, thank you. This first short presentation brings the perspective of ADR processing for medicines for human use in the post-marketing setting. So in the next 5 minutes, I will use 5 slides to talk you through 5 points that I would like you to keep in mind during the rest of the day. Eudravigilance, the human eudravigilance database, actually exists of 2 components, the EV post-marketing module and the EV clinical trial module. And I'm focusing now on the post-marketing module. So first of all, this module contains the spontaneous reports of suspected ADRs. And these are called spontaneous because they rely on the initiative of a patient, consumer, or healthcare professional to contact a pharmacovigilance center or MAH. And we call these suspected ADRs because they convey the suspicion of the reporter who takes the initiative to contact a pharmacovigilance center or a marketing authorization holder. A causal relationship doesn't need to be established. A suspicion is enough. And by international convention, when exchanging ICSRs of spontaneous ADR reports, these are regarded as having implied causality. And the mechanisms through which the spontaneous reports reach the NCAs or marketing authorization holders, these vary. And it makes quite a difference if a case is received using a structured web form or if you receive a phone call. Also, of course, there are the solicited cases, which derive from non-interventional studies and other organized data collection systems. So, in contrast to the passive nature of spontaneous reporting systems, this is a more active way of data gathering. And for these cases, doing a causality assessment is mandatory, and only those with a possible relationship should be submitted to eudravigilance. So, it's important to realize that there are actually 2 different data sets with fundamentally different methods of data collection and also different requirements of what needs to be submitted to eudravigilance. We stay with the spontaneous reporting schemes for a little bit while more. People often say that these are low in the hierarchy of evidence, and people refer to the limitations of spontaneous reporting systems, like underreporting or reporting bias, risk of duplicates, and also the variable quality of the reported data. I would rather call these characteristics, and that's also a word that we will use throughout the day, instead of limitations. Most of all, it's important to keep in mind the aim of a spontaneous reporting system, which is signaling and doing analysis on the data. And here I would also like to quote an old SIOMS 5 report from 2001, where it stated that these systems were not designed for or were intended to be a complete collection of every adverse event that occurs to every person taking every drug. More ICSRs don't necessarily lead to more potential signals. We move on to the third point. Actually, it's EU legislation and guidance that drives what is submitted by marketing authorization holders and NCAs. but also how it is submitted and the timelines. So when looking at the data set, it may be good to be aware of the impact of legislative changes on the data set, including how it impacts the quality. For example, the change from E to B R2 to E to B R3 has impacted several data elements. In R2, seriousness was captured at case event level, but in R3 it's captured at, in R2 it was case level, but in R3 it's event level. Also, a dataset may change because, for example, submission of non-serious EEA cases, which entered into force end of 2017 with the go live of the new ultravigilance system. The fourth point is about quality controlled processes. Stakeholders put a lot of effort in collecting ADR data and processing this as ICSRs. And I've used this picture to show how many detailed steps are actually taken into account when collecting information and processing ICSRs. And organizations have really robust procedures in place to do all of this work. And this is underpinned by hundreds of pages of guidance, and in GVP6, E2B-R3, an EU ICSR implementation guide, MEDRA points to consider. But nevertheless, we still have different practices. My last slide is actually more a question. What makes a case a good case? Is it if the data show that the adverse event that was reported as a myocardial infarction is really a myocardial infarction because it has the complete data, or if a causal relationship can be established, or if most of the E2B-R3 data elements have been populated, or if the case triggers further analysis. Also, it may be relevant to be aware that the details that may be needed may also depend on the context in which an ADR occurs, like a medication error or a pregnancy case. And I've put two examples here where the EMA, a good practice guide for medication errors, gives guidance for this specific situation. And there's also a recent publication from the IMI Conception on what needs to be collected for pregnancy cases. And for these special situations, is maybe good to realize that such details often don't have a dedicated structured data element in the E2B-R3 message. So with these 5 points, I've reached the end of my opening, and I give the floor back to you, Paolo. Thank you.
Thank you, Anya. Very nice presentation. I'm sure that it will trigger a lot of discussion later on. So, without other delay, I will pass the word to James Mount. James is a veterinarian and pharmacovigilance assessor at the Swedish Medical Product Agency and current chair of the CVMP Pharmacovigilance Working Party Veterinary. He is an expert in various EU groups, which include the HMA, AMA Veterinarian Data Hub and the Pilot Signal Management Expert Working Group. James, over to you.
Thank you very much. I'll share my slides. I can. Oops, jump to the end. Can you see my slides? Is everything okay? Up and running?
Yes, okay.
Thank you. Right, good morning, everybody. And I'd just like to start by thanking the organisers for inviting me to present, and also inviting the rest of the veterinary regulatory network to participate in this workshop today. I will give you a brief perspective from the veterinary side in relation to data quality and the issues we have, and in terms of adverse event reporting and the recording of these adverse events. And as Paolo said, I'm James Mountain, the current chair of the Pharmacovigilance Working Party and also veterinarian. So we'll move on to, if I click, has it moved on to the next slide? No, probably not. No, there. So the importance of data quality is reflected actually in the veterinary medicines regulations, which is relatively new, only came into implementation in 2021. And it's reflected in the fact that there's the mention of compliance with good pharmacovigilance practice in the regulation itself, and then also in the implementing regulations as the need for a quality management system for pharmacovigilance. And this specifically highlights the importance of collation of adverse events and data handling. So the existence of poor data quality in adverse events reports there's a number of consequences of this, and it's not just unique to the veterinary side, I don't think. The greatest consequence, of course, is that the ability to identify new risks related to the use of medicines will be impaired. And then when we're trying to analyze and derive these new risks, there's a negative effect on statistical methods that are used to derive these new risks, or so-called signals that we call them now. Then looking forward, particularly on the veterinary side, when we're looking for further sort of techniques to help us facilitate identifying these new signals, there may be an impact on the efficient implementation of new techniques such as data mining and these other forms of AI techniques. And in my opinion, I think on the veterinary side, we need to have at a general basis, have a focus improvement on data stewardship. The poor data quality impacts all stakeholders, and it's especially the marketing authorization holders, purely for the fact that now they're obliged to signal detect in the European collective adverse event database. And also, one important aspect is that the actual general public can also be impacted by poor data quality, as this data is actually publicly available in the public portal of the European database, and that's purely through transparency, and I'll provide the link for people that are interested in looking at that. The Pharmacovigilance Working Party, which I have the privilege of chairing at the moment, has on numerous occasions discussed the impact of data quality and continuously also discussed related issues to data quality. And in 2022, there was a survey of members, and it revealed, amongst other things, the sort of top 5 observed data quality issues that are observed regularly in adverse event reports. And I'm not going to go into the depths of these now because due to time constraints, but I think Catherine Sherman, who's from the German agency, will be delving into some of these aspects in more detail, I believe, later on today. And this is just to finish off. Maintaining data quality is a collaborative effort and a collective effort of all stakeholders, and that goes with veterinary pharmacovigilance as a whole, as in we all have a common goal to maintain efficient and safe medicines for use. On the regulator side, we have currently a multifaceted approach to trying to monitor and prevent data quality issues, and I've tried to outline some of these here, and it's not an exhaustive list, but this includes the quality review of individual reports, and this is done by some competent authorities, but not all. This is a very labour-intensive effort and also heavily dependent on resources at a member state level, of course. But it's a very effective strategy if you do it on an individual basis. EMA itself have invested quite a lot into algorithms in the pharmacovigilance database, and that is the Eurovigilance veterinary we're talking about here. The focus primarily is on duplicate detection, but then it is envisaged that going forward there will be an expansion of the current business rules in the system to help us to combat issues in the future. On the veterinary side, we have a relatively regular dialogue with our veterinary stakeholders and discuss quality issues in reports. And some of these discussions often result in updates of coding instructions or other relevant guidance. So it's a form of data stewardship. And these discussion forums are a good way to be open and also to follow up on issues that have arisen previously. And then a very recent initiative that we've sort of embarked on is to have a regular discussion with our inspectors and the pharmacovigilance experts. And this is at an EU level. And the aim with this forum is to have an opportunity to discuss quality issues and also to have transfer information. And it's proven mutually beneficial at the moment for both parties involved there. So we're considering to continue that process. But that completes my presentation, and I hopefully gives you just a brief introduction to an overview of the veterinary side. And without delaying proceedings further, then I hand back to Paolo. And thank you very much.
Thank you, James. We will come back as well during the discussion on this point that you made, I think. Thanks a lot.
Thank you.
Now I would like to ask Elke Stahl to start her opening remark. Elke is a senior expert in clinical trials at the German National Competent Authority, BfArM. She is leading the CTCG safety subgroup since 2009. The group developed the best practice in safety surveillance in a clinical trial, as well as coordinate, discuss emerging safety issues. She joined the ACT EU PA9 as coordinator lately, and she has been long-term product owner and subject matter expert in the CTIS and BI tool focusing on safety. Elke.
Thanks, Paolo, for kind introduction. I'll start sharing my slides. Oh, yep. I would like to give you, oops. Okay, hope you can see them. Introduction to adverse drug reaction reporting and the impact on clinical trials. I think the goal for clinical trials, no, you can't see all. The goal is really to characterize active substance, the medical product in the respective population in a controlled setting. And as of the clinical trial regulation, we have different responsibilities. So the sponsors have to ensure participants' positive benefit risks on a continuously basis. So they need to monitor upcoming safety information signals and have an adequate risk management. In addition, there are a couple of safety reporting requirements in the regulation, which is nothing new in comparison to the clinical trial directive. So you need to report SUSAs, an annual safety report, and notification if something is affecting the benefit risks, urgent safety measure, temporary hold, and so on. In addition, what is new in the regulation is that there are some exceptions possible as of the protocol for the recording and reporting from the investigator to the sponsor and sponsor to the agencies. Although the responsibilities of us member states is to have a safety surveillance, especially for SUSAs and annual safety reports. But to tell you the truth, we look also on notifications, all what is relevant for safety, and check if there's an adequate risk management. So the impact here is really on safety of participants in clinical trials, on a balanced benefit risk, also in clinical trials and later on in the authorized setting. And that's a little bit sketching where we are. We are early on. So we are in the beginning of the investigation of an active substance in human. And what is important here is really the interaction between the participants in clinical trial and the investigator. The investigators are in charge of the good health of the participants, and they need to really monitor adverse events, record them, and check for causality and seriousness. And if we have a good quality from the beginning, we will have a good quality in the reports, the narrative reports, recorded and reported to the sponsor. Sponsors, sure, as you have seen, will analyze and draw up their conclusion, checking the causality. So if it's an adverse reaction, the seriousness. If it's serious and unexpected, then it needs to be reported expeditiously in the eurovigilance. And here it is the eurovigilance clinical trial model. For some, also the PM model, but that's more for the auxiliary medical products. And in addition to this continuous monitoring reporting, there's a cumulative report expected on an annual basis in format of the ICH E2F DSUR, the annual safety report. That's now reported via CITES, and everything else is reported via CITES, the notifications, for example. And member states, we take a look at the annual safety report, sure, but we also continuously monitoring SUSAs quite frequently, and checking if all is fine and taken care of. So give you a bit of insight on our experiences here is that for the annual safety report, we all need to check if there are changes between the reporting periods of a year with regard to frequencies, the issues identified, the seriousness, if some new potential issues coming up, some old could be closed or why they could be closed, and take a look on the cumulative tables. What is more work for us, to be honest, is to really dig out new information in the SUSE. So we're driving out reports from the ultravigilance database and check if there's something popping up which is relevant to have risk mitigation, relevant to monitor, to be aware of. And here it's important that it's really unexpected, as of the reference safety information where it's checked against, that the causality is given, so it's neither due to the disease or the different diagnosis is needed here, and it's also not to the co-medication only. It could be to the combination of both, for sure. We look for new cases, not if new cases are really follow-up cases or merged follow-up cases. We need a clear category here of the issue, the preferred term. And so we have good quality in the structured data and maybe a bit more structured data than we have currently. So we need to get good reports out of ultravigilance. And in addition, we later on look on the narratives, and here we really need complete case descriptions, sufficient detail to know when was the onsetting, other co-medication, co-funding factors, risk factors, to identify relevant cases. And to tell you the truth, if it's for a marketing authorization in specific disease like oncology, it's getting intense to dig out. If we need for more information, or if it's like not used in the authorized population but off-label. So it's important for us to have high quality, to analyze the reports we get, and also for the further use, because this is the basis of the safety profile. It's a basis of decision later on. But for us currently, it's really to ensure that the patients are fine. And thanks, and handing back. Thank you.
Many thanks, Elke. After these initial opening remarks, where we saw remarks from human medicine, vet medicine, and in particular for pre-authorization phase, we will move now to the first session where Anna Cocchino will give us an overview of the recently published European Data Quality Framework. Anna is a medical doctor working in data analytics in the Data Analytics and Method Task Force at the EMEA. She's leading activities around strengthening the role of real-world evidence in medicine regulation with a focus on data discoverability and data quality. And she's involved in ICH international activity like MEDRA Management Committee, of which she's a member. And previously, she had 10 years of experience in the area of pharmacovigilance, leading on activities related to the implementation and guidance of medicinal product dictionary in the context of adverse drug reaction reporting and ultravigilance. Anna, please.
Thank you, Paolo. I hope you can hear me. Yes. It is my pleasure and also my challenge this morning to try to run you through the data quality framework on what we have done during the last 2-3 years in this work, and to try to convey some of the concepts that are inside. I hope I'll be successful in doing so. But starting from the very beginning, I will try to go a little bit through the background and scope of the EU data quality framework activities, why we have done what we have done, and all the activities around it, the timeline, the steps that we have taken so far briefly, and what we plan to do next. It will be a little going in details, looking in dimensions and subdimensions of what we defined as measurements for data quality, briefly touching on maturity models, and at the end, perhaps some points to consider specific to the topic of today's workshop. So the background of this work is tying back to the Big Data Steering Group, decided, and being a HMAMA joint working group, this group decided that its direction to for the medicine regulation to be more data driven, and in being so, we have been tasked to develop a data quality framework. You can see on the right there are a lot of recommendations that the big data steering group put forward, such as improved data discoverability and so on and so forth. We are focusing in this context on the data quality and representativeness. After the setup of the methodological working party last year, I think, of the CHMP, the sponsorship of this work became co-shared between these two groups. So we both have a sharing from methodological working party and also a big data steering group sponsorship. And eventually this collaboration ended up with our guideline being a CHMP approved guideline published as such. In drafting this document, there has been a close collaboration with experts from various regulatory fields. We talked to people in quality, in preclinical, veterinary colleagues in ADR. We have widely consulted, we had public consultations, and I'll be a bit more detailed on that in a second. We tried to draft a document that covers all aspects of data quality in this medicine regulation purpose. So the concepts are applicable to all data sources, and that perhaps was the easy part, maybe, because now we are tasked to go deeper, to take this data quality framework that sets up the ground for this work and to take it deeper in various contexts. So we have ongoing work on the real world data domain, and this is the somewhat kickoff of the work on the adverse drug reaction. Bit of history. So I was saying that in 2022 we start off with drafting this EU data quality framework that I'm presenting now. It went through some phases that are somewhat similar for every use case that we do. So we went into a landscape analysis of what data quality frameworks exist and similar initiatives. We had a working group that I mentioned a bit earlier, veterinary, preclinical, ADR, and so on, that looked at what we are doing from the point of view of their expertise. We had a multi-stakeholder workshop, just as this one, back in 2022, where we gathered people's thoughts and issues with data quality to better address them. We went for a public consultation towards the end of the year in 2022. In the next year, we addressed these comments. We have went through another round of consultation with CHMP, and towards the end of the year, we got to a point where we had a document that was accepted and approved by the CHMP, and that was published. As I was saying, we start doing the deep dive. So then we started doing a deep dive in the real world data. We had a similar workshop with the experts in the field of real world data sources, and back in June last year, and we produced, based on that output, a first draft of data quality framework in the real world field. It's still early days, but we have been progressing. Same goes for the ADR topic. We're early days. We have done a bit of research. We've done some preparatory work, and now we are at the point of a multi-stakeholder workshop for ADR data. And what comes next is to continue in the same sort of workflow. We will be preparing a first draft sometime later this year, and we'll think through a consultation process. It's still early for ADR to really say what steps and timelines and so on, but we are started the ball rolling. And in terms for the data quality framework in real world, as some might be interested, we're looking into having first a regulatory network consultation of this first draft, a redrafting, and then we will proceed with the public consultation on this document. Timelines yet to be confirmed. So what is the scope of this document? So I have in various points to this presentation, I have links towards it, so you can open it, have it a look at yourself. It's a 30-40 pages document, pretty dense, but we like to think quite extensive, covers the topic pretty well. Well, it seeks to address the topic of characterizing data quality on any given data set with a focus on the medicine regulation. Naturally, being somewhat high level, then also the concepts will be defined at that level of data quality in general. So it sets out concept definitions, metrics, and proposed in the end a set of maturity models to describe the quality of data sets. We have noticed, and we're not the first to notice this, in a landscape analysis at the beginning, that the definitions for various terms vary in data quality, and each field has a bit of a different flavor of the same thing, and sometimes completely different definitions for exactly the same concept. Hence, we set out to try to harmonize all of this work and to have a starting point, to have a common terminology dictionary and some ground to work from, which is a framework. So we aimed at to provide a coherent basis to work on further. This is a one sort of slide look at the concepts of the main concepts that you would have inside the data quality framework. Naturally, I won't try to go through all of them. They are at length described in there. I will probably focus in the next, say, 5 minutes or so on the bottom part of this slide. So I will try to go through the dimension of data quality listed here: reliability, coherence, extensiveness, quality, and timeliness, and what we defined as sub-dimension of these dimensions. So looking a little deeper in them. As I describe them, I would encourage to think of how this works for your expertise, for your domain, not necessarily focusing on the terminology, but thinking of how would this help me, how I would express this in the data that I'm expert of. It might make it a bit more meaningful and more clear if you think it like that. We have split the data quality aspects that we call determinants in 3 different categories, and based on these categories, then the maturity model of data are being described. So we talk about things that are foundational, so talk about how the data is being generated, the process to generate, to collect it, and to expose the data. Then it's the middle level of intrinsic determinants, which talk about aspects that are inherent to a specific data set, so specific to the data set as an entity. And then there are quality determinants that are question specific, so they are driven by addressing a specific research question or a specific use case, if you'd like. And these are the main dimension in a blink that we will look at. So we're looking at things like is reliability. So is the data correct? Is it representing what it's meant to represent? And I'll be a bit more detailed in a second. Then we're talking extensiveness. How much data is there? Is it covering what I would like to cover for my use case? Coherence. Can data be analyzed as a whole? We're talking timeliness, if we have the data available at the time that we need it, and relevance, whether the data is the type that I need for my work. So, speaking on of reliability, I was saying we're talking about a dimension that looks at to what degree are data corresponding to reality. It goes further to look a bit closer. So it gets in describing the sub-dimension that could be used to describe that. So precision can be defined as degrees of approximation by which data represents reality. And it's an example here to make it a bit more meaningful. So say the age of a person that could be reported in years or in months. So how precise is your data? Then we define accuracy as the discrepancy between data and reality. So if you have, say, downright an error, a random error in measuring, say, weight, or you have a systematic error by measuring your patient fully clothed, for example. So accuracy. And lastly, we'll have a sub-dimension named plausibility that is somewhat of a proxy for accuracy, a way of identifying issues with the accuracy and is defined as the likelihood of some information to be true. So say outliers, weight of more than this amount of kilogram, it's likely an error. So that's the picture on reliability. The next dimension, extensiveness. It looks into how much data we have, whether, and specifically because we're thinking fit for purpose and we're thinking regulatory information, so not necessarily trying to characterize the whole world, but our purpose in mind. It defines the quality in the sense of extensiveness that covers how sufficient the data are for fit for purpose. It has two subdimensions. One refers completeness, refers to more or less missingness. So would you, if you have the amount of information that you have with respect to the total information that you could have, so it's completeness, missing things. And then the second one is coverage, that talks about the amount of information you have with respect to the whole world. So say how much of the population of a given country a data set has. Third one, timeliness, as the availability of the data at the right time for regulatory decision making. Probably this dimension and the next one are a little less, are more under control of this particular use case in adverse drug reaction. So looking at currency, whether the aspect of timelines that considers how fresh the data are. So do you have it when you need it? So answers that question. Relevance is defined as the extent to which a data set presents data elements useful to answer a research question. Again, just like timeliness, it's likely not so much of an issue for a primary use of data where we define what we collect. But for completeness, it describes if the data set presents the kind of values that we need to address a specific question. And in general, when we define the process from the beginning, we would collect those data, so it's not necessarily an issue. Perhaps more interesting for this topic is coherence and its subdimension. That's coherence would express how different parts of an overall data set are consistent in representing them, what they mean. So they're split in four kind of subdimensions, and they're looking at format, whether data is expressed in the same way throughout the data set. So for instance, different dates that could be expressed in different way. There's structural coherence, when entities identified in the set should be identified in the same way throughout the set. So thinking, knowing, being able to follow your, say, report or patient through various bits of your data. Semantical coherence, that's a little bit different in the sense of if the value means the same thing, it is the same one, but does it mean the same thing to various parts of your data set? And lastly, but not leastly, uniqueness, to make sure that the information is not duplicated and appears in a data set once. All right. I will not, since I have very little time and I would like to not overload with information, I'll touch more high level on these data quality maturity models, just to explain the concept. What it does, so the data quality framework defines all the dimensions as I have run you through in the last, say, 5 minutes. What it does then is to take these elements and put them in maturity models to try to define characterization method for what we have explored in our data. So goes in the foundational sort of determinants and ranks them from, say, documented to automated, and describes in detail what each means. Documented being to have your data quality and data processes in general well documented with some metadata, and then it gets more and more advanced. So you have more metadata in a formalized process. You follow industry standard or automated. The whole process is automated in terms of documenting the foundational determinants. Second category, intrinsic. So data quality that is intrinsic to a data set goes from a very basic level with very basic data quality and grows up to having data elements specific to data quality in the metadata, having a standard applied, automated throughout the system and all the way to feedback to the collection part to improve the data quality through its metadata. So again, last level being somewhat of an aspirational level. And last one, question specific means that the data is already set up in such a way that it can answer some questions in a predetermined way. So, and this can be growing in levels from ad hoc. You can get the answer to a question on an ad hoc basis. You can have some domains defined that give you some guidance on how exactly you would get for a particular domain of questions an answer, or it can be question defined and you'd have the answer directly in a half automated way, if you like, on predefined question. There's a lot. These points to consider naturally coming from us as data quality, from our data quality work, and what we've seen in our past use cases being highlighted. This workshop, I'm sure, will highlight a ton more. But to start from somewhere, I can tell you that it seems always an issue to think also outside the EU, the non-EU data sources in alignment with other data quality frameworks and the opportunities for international convergence. There's a heterogeneity of different EU systems. I think that's true for any system, what we'll see today. There's a need for this work to result in guidance or tools to help implement the maturity model for the different regulatory use cases. So to take all that I've shown you all the way to a level where we have, say, a checklist or a scorecard, something that can be used by not experts in the end. This is our final goal, to have something that is usable. So that's something to have in mind. We should think also of the implication for system owners and data submitters. This depends on the use case, but that's something to always keep in mind. That not always the measurement I have went at length in describing measurements and percentage and so on, but that's not necessarily always needed for a given use case. Sometimes just descriptive documentation is sufficient for some use cases. And of course, the difference between this particular ADR use case as primary use of data uses data that was initially designed for this purpose. So that will have its own particularities that are different from, say, secondary use of data, where ADR can still be considered. So it might worth keeping also an eye out for that. Closing with just a few points. So what I'd like to, I realize it's a rather dense concept and it won't necessarily be absorbed just like that. But what I wanted to say today is that we have a EU data quality framework. There's a guideline, has been published. You can access it on the web, and it has been adopted by CHMP, and we would like to take it forward. There's overarching concepts, so don't be put off. Sometimes people see the examples being more, I don't know, not necessarily in their topic and thinking it wouldn't apply to the topic. That's not the case. Concepts are overarching, so keep that in mind, that it's a good document to use wherever your expertise lies. The drafting process is just began, so there'll be a long row of other stakeholder consultation communication, so bear with us on that. And for any other questions, comments, proposal, whatever idea you might have, you can reach out to our mailbox on data quality framework, and we'll be happy to receive any input. And thank you for your attention.
Many thanks, Anna. Anya, are you there?
Yes, of course, Paolo.
Perfect. So would you like to take over the chairmanship?
Okay, thank you. So now that we have been introduced to the data quality framework, we are moving on to the next session, which provides a regulatory overview of the ADR data quality activities. And the nice thing is that we have both EMA and NCA perspective, as well as a human and veterinary view. We start with a presentation from EMA, combining human and veterinary, Then we move on to a human NCA view, and then we have a veterinary NCA point of view. For those of you who have been joining later, there's also some time allocated for Q&As. And if you wish to speak or ask a question, please wait until the time allocated for the Q&As. Raise your hand for those joining in WebEx, or put something in the chat. Let me now introduce the first EMA speakers in this session, which are Tom Paternoster and Laura Descalzo. And Tom has worked in pharmacovigilance for the last 23 years, starting with MHRA, then moving to industry for 2 years, and he has been at EMA for almost 20 years. And the main focus of his work is duplicate management, ICSR data quality, and of course, the medical literature monitoring service. Now, Laura brings the veterinary point of view. She is a biologist working in the veterinary division at EMA with 20 years of experience in the area of pharmacovig, leading on activities related to the implementation of the Union Pharmacovigilance Database. Laura and Tom, the floor is yours.
Thank you very much, Anya. So I should be, oh, my slides aren't working. Sorry, thank you. I was using the arrows, strange. Okay, thank you. We'll have a presentation now on the covering the guidance and indeed legislation for humanary and veterinary ADRs and AEs, the standards, EMA data quality activities, and EMA uses of ADR data. Now, because this is rather an expert audience in this workshop, I'll be going quite quickly through the legislation and guidance and standards, because I'm sure that everyone here will be aware of them. And then we'll have more time to discuss the actual data quality activities. So, legislation and guidance. Human ADRs are covered by Regulation 726/2004, Directive 2001/83/EC, which covers more for MAHs and NCAs, and Commission Implementing Regulation 520/2012, and Regulation 536/2014, the Clinical Trials Regulation. Veterinary AEs and data quality are covered by Regulation 2019/6 and Commission Implementing Regulation 2021/1281. For human ADR data quality, there's an awful lot of guidance, as I'm sure everyone knows. There's a good pharmacovigilance practices, which are a set of measures drawn up to facilitate performance of PV in the EU, and modules 1, 3, 4 and 6, including addendum 1 on duplicates, are all very directly related to ADR reporting and data quality. SUSAR guidance is provided in CT3 and not mentioned here on the slide, but we also of course have reaction coding guidance, as Anya mentioned, from MEDRA points to consider term selection document. For veterinary ADR AE data quality, it's important to note for vets it's more related to adverse events, not necessarily drug reactions. There's the VGVP for collection and recording suspected adverse events for veterinary medicinal products, very similar to GVP Module 6. That includes the appendix, the EVV best practice guide. Also there's pharmacovig systems on GVP and controls and pharmacovig inspections. So very similar human and veterinary guidances. Standards, there's two main standards organisations whose standards govern the pharmacovig and ADR reports in the EEA, and that's the ICH, International Council for Harmonisation, and the ISO, International Standards Organisations. There are, of course, other related organisations whose work has contributed to the development of standards, including CIOMS. Everyone, I'm sure, remembers fondly the classic CIOMS 1 form, and HL7, who are creating a FIRE version of ICSRs. So, ICH standards directly related to ADR reporting include M1, E2A, E2B. Everyone will be very aware of that, E2D of course, and also E6 and E19 relate to ADR reporting as well. the ISO standards, there's health informatics for ICSRs, so human pharmaceutical reporting requirements for ICSRs, and there are 5 standards related to medicinal products under the heading of IDMP. They're not yet implemented. Presently, XEVMPD is a product standard, and EDQM is for dose forms, units, and routes of administration. The ISO standards will be coming in the future. For veterinary AE reports, there's the VICH, and there's ISO, and also HL7 is a relevant standards organisation for veterinary reports. And there are 4 VICH standards directly related to AE reporting, which you can see here on your screen, and copies of the slides will be made available afterwards if people want to have these. So I'll now move on to the EMA ADR data quality activities. Now, the data in EV is highly heterogeneous. There's over 99.7% of our data is submitted by 3,600 different MAHs, NCAs and sponsors. And the other 0.3% is submitted by about 1,400 other MAHs and sponsors. So that's quite a wide variety of organisations, each with their own different standards. Although one would hope here, following the guidance and standards well, the agency is responsible for operating procedures that ensure the quality and integrity of the information collected in EV. That's Article 24.3 of Regulation 726/2004, in collaboration with NCAs and MAHs, the organisations which send the ICSRs to EV. And to help stakeholders understand the processes the agency operates and to improve their own data quality, EMA published the detailed guide regarding the due diligence data management activities by the EMA. Now, the systematic approach to improving data quality by the agency can be summarised on the next couple of slides. Consistent, complete, correct and well-structured information submitted in ICSRs and to the XEVMPD is one area of the operation of a quality system and is very much necessary to perform PV monitoring and evaluation activities, including signal detection. These quality assurance activities can be summarised as followed. We've got the adherence to pharmacovigilance legislation and regulatory guidance, offering hands-on training courses and the provision of e-learning modules, pre-production testing with organisations preparing for the electronic submission of ICSRs to EV, the application of business rules in EV to assist an automatic validation against predefined parameters, duplicate detection system and management to address duplicated information of cases submitted by the same or different sender organisations, the validation of data submitted to the XEVMPD in accordance with Article 57, 2, automatic and manual reclassification of reported suspect or interacting medicinal product information against the XEVMPD to allow for reliable data retrieval and analysis, EV post-validation database level checks to assess case validity and report retransmissions, the periodic review of ICSRs submitted by organisations to EV based on data sampling, so that's in-depth, individual cases, compliance monitoring based on reporting timelines set out in the PV pharmaceutical legislation, quality audits as well, and the conduct of pharmacovigilance inspections by competent authorities in member states. And these QA activities are summarised on the next slide, and the document I linked to earlier covers points C to I in great detail. So the next slide shows what I was talking about. You've got at the top guidance and training, then over to the right, testing, business rules, data quality and compliance and duplicate detection. And you can see how these circles are in kind of 3 groups with overlapping parts, and the overlapping circles represent overlapping processes which rely significantly on each other. Then coming round the clock, you've got the XEVMPD, medicinal product reclassification, and then lastly, access to EV data. Although that brownish circle with the access to EV data should also have an overlap with business rules as well, because any cases rejected aren't accessible to external parties. So pre-production testing. Cases and products transmitted to EV can be created in one of two ways. You can either use your own database or that of a contract research organisation, or you can use EV Web. If you're using your own database, then before an organisation is granted permission to transmit to EV, they have to prove that their database is compliant with the E2B standards. And back in the day it included R2, but now it's only R3. This is done through the perfect transmission of test cases devised by EMA to ensure that every data field and case type is checked. So we've got about a dozen cases you have to use for your test cases, and we verify that you can load them into your database and then transmit them as they should be, and that the data is in the correct fields and of the correct type. If the organisation is using EV Web, then we know that the database is fine because we built it, but at least one member of the organisation staff has to take the face-to-face EV training course and pass a final exam as well, which is quite a strenuous exam. So there we're testing the users. Business rules. Now, demonstrating compliance with the standards during testing does not mean that all cases created using a particular dataset will be correct. Ergo, every ICSR is automatically assessed by the EV parsers against the business rules before being loaded into EV, and then an acknowledgement message is returned to the sender. Business rules cover the following: population of mandatory fields, including data type and field length, logical population of fields, so you can't have future dates, contextual mandating of fields. For example, if a reaction outcome is fatal, then it's mandatory to have a death section. If an ICSR fails the business rules, it's excluded from all PV activities and queries. Additionally, the expedited compliance clock doesn't stop for your case until you've submitted a successfully loaded ICSR. Now, duplicate detection. Duplicate cases in EV can lead to false positive signals, wastes assessors' time, and it can also mask true signals by inflating the baseline of the ROR. Therefore, the agency detects and merges identified duplicate cases to ensure that each ADR is counted once in EVDAS. The process can be summarised as follows. Our algorithms assess all the cases in the database for potential duplication. The potential duplicates are then assessed by our human assessors, and if duplication is confirmed, a master case is made and transmitted to EV. And that master case becomes the case version for pharmacovigilance. The other cases underneath are still live for follow-up from the original senders and for audit trail purposes, but then wouldn't be viewed in pharmacovigilance queries. Reclassification of products and substances reported in ICSRs. Now this is vital. Drug and substance names in ICSRs are not based on a controlled vocabulary. They're free text, and only the presence of any text in the drug name field, that's GK22, is sufficient for a case to pass business rules. You could put A and that's it, that's adequate. In order to provide usable pharmacovigilance data and to make data available to MAHs and the public, this free text data needs to be first normalised and reclassified against data from the XEVMPD and then subsequently grouped by active substance. Our system attempts automatic reclassification overnight. So at 6 o'clock, the EV shuts for a moment. The parsers run against all the cases which have arrived from 6 o'clock the previous day until 6 o'clock that day, and then attempts to reclassify. And over 99% of the drugs and substances in ICSRs are successfully automatically reclassified. If you write paracetamol and spell it correctly, that's automatically reclassified. But if someone makes a typo, rapacetamol for example, then it might not be automatically reclassified and it gets sent for manual reclassification. However, the way our system works is that once rapacetamol has been recoded against paracetamol, then all future rapacetamols will automatically be reclassified, which is why over time our success rate of automatic reclassification increases all the time. We have database level queries as well. Now, GVP Module 6 sets out 4 minimum criteria for a case to be valid. You need one or more identifiable reporter. You need one single identifiable patient. You need one or more suspect substance or medicinal product. And you need one or more suspected adverse reaction. But every one of these elements can be populated in order to make a case appear valid, even when the sender lacks relevant information to make it truly valid. This can mask true signals by inflating the denominator used in the ROR and similar calculations. So, for example, if you don't have any demographics about the patient, you could populate unknown in the initials, or just UNK typed in, and we don't know if you don't know or if it's Umberto Neil Knowles, and so our system has to accept it, but then we try to identify them. Or indeed the MEDRA LLT just adverse event or adverse event NOS or even no adverse event can be populated. It's a valid MEDRA LLT, so again the system accepts it. EMA has created queries to identify such cases within EV. Now, this information is presently mostly used by us to target further assessments or in discussions with our colleagues in inspections, but in the future we plan to share it with sender organisations. Periodic individual case review. Now, in addition to the database level queries, we perform periodic reviews of samples of ICSRs based on the parameters for the content of an ICSR as set out in Article 28 of Commission Implementing Regulation 520/2012. And the process can be summarised as follows. We select between 10 and 20 senders each month. And remember, we have 5,000 or so senders a year. So there's a lot of senders to get through. From the senders, we pick typically 10 to 25 recent cases, although if there's a particular request, we may do more cases for a particular sender. And we include a variety of different case types, fatal cases, SUSARs, spontaneous cases, some literature cases, and then we prepare a report. In most cases, we need to base it on the narrative to see if the structuring is done correctly. If it's a literature case, we can go and try to find the literature article and see if the whole case was correctly structured. And our findings are classified as high impact, medium impact, and low impact, depending on the effect of the error on pharmacovigilance activities. So a high impact affects signal detection directly, wrong drug, wrong reaction. Medium affects the most common signal analyses, for example, errors in patient history or patient demographics, and low are mainly administrative or typographical errors. Once we've sent the report to the sender, we ask them to fix any cases, implement CAPAs, and comment on the report. And because we're basing it on the narrative, because we don't see the source documentation, it may be that what looks like erroneous data at first sight is actually justified and not an error, in which case we're very happy to amend the report. So if you receive such a report, please do review and feel free to challenge any comments we've made. Now, additionally, consistent, complete, correct and well-structured information in ICSRs and the XEVMPD is one area of the operation of a quality system and is necessary to perform PV monitoring and evaluation activities, including signal detection. Oh, sorry, this is actually for the veterinary side. I shall hand over to my colleague Laura Descalzo, who will take it from here. Laura.
Thank you, Tom. If you could please go to the next slide. Firstly, I should mention that the data quality activities and tools are not yet as mature on the ultravigilance veterinary side as they are on the human side, because we are still developing some of the systems and processes. But on the next slides, I will quickly run you through some of the existing activities. So, for example, as mentioned by Tom, we've got like a mirroring process for pre-production testing, and this is related to organizations that use their own database. So we send them a set of reports that they need to transmit to prove that their database is compliant with the VIC8 standards. Next slide, please. We also have business rules implemented in EVVET, which They are assessed by the EV parsers, and they cover more or less the same areas that Tom explained before, such as the population of the mandatory field and also the logical population of the fields, the same no future dates, contextual text, and so on. And if an adverse event report fails the business rules, then it's not used for data analysis. We also have implemented the process of reclassification of products and substances, because the same as on EV Human, the product and substance names can be submitted either as controlled vocabulary coming from the Union product database or as free text. So hence, if the report contains free text, we also attempt to automatically reclassify overnight. And if this fails, we also have a manual reclassification process. And then we also have a duplicate detection tool that has been recently implemented. At the moment, we have what is called a minimum viable product, and we are currently developing new functionality. And I will briefly summarize the process. So there is an algorithm that assesses all the cases in the database for potential duplication. And then those duplicates are manually assessed. And if the duplication is confirmed, one of the reports is designated as the principal report, and the other report is marked as the duplicate. And the principal report is the version of the case used for analysis. However, all the underlying duplicates also remain available in the database for audit purposes. And there are some processes that are still being defined, like, for example, follow-up to duplicate reports. And Tom, over to you. Thank you.
Thank you, Laura. I now, on our last slide, discuss the EMA uses of ADR data. Very, very briefly, because we're pressed for time. So human ADR reports are used in the following ways by EMA. We use them obviously for signal detection and analysis, which is their primary purpose, for compliance monitoring, for the publication of data, for creation of non-serious line listings for PSUR review, and also we've recently started using them to set the submission frequency of PSURs. Veterinary AE reports are used for signal detection and analysis, compliance monitoring, and the publication of data. And that's the end of our presentation. Thank you. Over to you, Anya.
Yeah, thank you, Tom and Laura, for this interesting presentation. It's interesting to see what the EMA is doing, being at the end of the whole process as a receiver of all those data. But now we move on to one of the senders of ADR data, an NCA. And I'm happy that we have Elena with us, and from the Austrian agency. Elena studied biology, holds a PhD in natural sciences from the University in Karlsruhe, and she worked several years in pharmaceutical industry as a regional safety lead before she joined the Austrian agency in 2021. And since then, she completed training as pharmacovigilance inspector and is performing pharmacovigilance system inspections for both human and veterinary medicines. Elena, the floor is yours.
Thank you for this introduction. Can you see my slides?
Yes, we can.
Yes,
thank you.
Thank you. So I will provide today the view of the National Competent Authority regarding the uses of the ADR data and data quality activities that we have. Our competent authority is responsible for both human and veterinary medicinal products, but I will just cover the human part. I will talk today about our pharmacovigilance activities, how we use the ADR data, about what quality activities we have, and also a little bit about the issues that we identify to provide you a feeling of what we can identify with those activities. I will not go into the legal requirements much because Tom covered that really nicely. So as per legal requirement, we as the National Competent Authority operate a pharmacovigilance system that covers those main topics, ICSR and signal management, PSURs, RMP management, communication, and inspections. That's where I come in. And my point of view, of course, is a little bit shifted towards the inspections, and we work quite closely with ICSR and signal management assessors and at our health authority. So starting with the point how we actually use the ADR data, we have routine pharmacovigilance activities. We have set up the national ADR collection system where the healthcare professionals and the patients can report directly to us. To provide you a little bit of a feeling how much data that is. So Austria is relatively a small country. We receive on a normal year somewhere around 15,000 cases. The majority of those cases are reported by the market and authorization holders, and we receive those through Vigilance, and somewhere between 15 and 20% of the cases that we receive are reported directly to us. And that's around 2,000 to 2,500 cases per year. Of course, we had 2021 and 2022 where the situation was radically different. For example, in 2022, we received almost 125,000 cases in Austria, and the amount of cases that were reported through the by MAHs actually didn't change that much. That means that more than 100,000 cases, more than 90% of these cases were received directly at the health authority, which was, of course, a huge load and huge work done by our colleagues at the case processing. So what we do with this data, we have a responsibility to monitor the data on the national level in terms of signal management, of benefit risk evaluation, and this data also used in some other issues. Mostly this is statistical data. There are some political inquiries and so on. And then for the substances that Austria is responsible for, the reference member states, we perform signal detection and vigilance. Regarding the data quality issues, so for these are several parts for the case processing. Of course, we actually add to the data quality, and we try to maintain our system so that the processes are set up correctly. We have our training that the assessors have to perform before they start case processing. And during the case processing, we perform duplicate check, the quality checks, and we have structured follow-up process where we collect the information that is missing, and also our assessors, for example, identify the events of interest, and they might develop a questionnaire to collect more meaningful data in process of follow-up. And then we identify the data quality issues more or less on case-by-case basis during our activities in monitoring of adverse drug reactions and signal detection. And then we have quality checks at the MAH level during our inspections in GVP and GCP inspection. So regarding the issues that my colleagues see a lot during ADR monitoring, the first that comes to mind is, of course, that we have a lot of cases that are not complete. We have very few well-documented cases, and this makes the validation of signals very difficult. Then we have issue that I just described here as noise. There are several different issues that are duplicates, some retransmission of cases without additional data by the organizations, have the non-valid cases that are reported to user vigilance, and also we have cases with a lot of non-relevant information inside that is that you need to work through, but it doesn't add any meaningful information for actual assessment of the case. Then we have an issue where the data are not coded in structured fields and only provided in narrative of the cases. And this makes of course, the working with the data in systematic way quite difficult, because you have to actually read through all the narrative to understand what data are there and what not. Then we have, of course, mistakes in coding and some other examples where the references are not provided and so on. And this information we do receive as inspectors as well, and it does influence our EV inspection program. So we have responsibility to perform inspections, and most of inspection in Austria are routine inspection. We have a certain interval of 4 years for routine inspections. We have our national program combines the inspections where we have a supervisory authority. Those are the organization where BSMFs are in Austria, and they have centrally authorized products. Also the companies where BSMF is in Austria and they do not have centrally authorized products, and then we perform some affiliate inspections. So the signals that we get from the assessors, also from other inspectorates, and some of the activities that Tom mentioned previously from EMA, they can either trigger an inspection or shorten an interval or shift focus of our inspection, which happens very often. To provide you a little bit of statistics, so we have in Austria on a human side around a little more than 200 PV systems, 70 plus of them are with BSMF in Austria. In 5 cases, we have supervisory authority, the companies with centrally authorized products. And then we have around 20 PV systems on that side with 1 caps. We aim to perform something between 25 and 35 inspections per year, depending on how big those inspections are, how much resources we have. So for example, in 23, we performed 34 inspections, 31 of them was on the human side. To provide you a little bit of the statistics on the findings. So this is the data for 2022 and 23. And the first graph just shows the general amount of findings that we see in those inspections. And the second graph is normalized per inspection. So as you see, we have quite a stable amount of findings that we detect during our inspections, somewhere between 5 and 6 findings per inspection. We did see some increase of critical findings in 2023. The reason being mostly that we re-inspected several companies that had previously not did so well in previous inspection, let's say. And here I try to summarize the findings that we have purely connected with the data quality for ADRs. So they don't, I didn't put here the data for other processes that can influence the data quality. So I tried to isolate just the data quality issues that we have. That's again the data for 2022 and 23. So we had 16 findings in 2022 and 26 in 2023, with majority of findings are minor, but we had quite some major and critical issues as well. And this doesn't necessarily mean that the data quality became worse in 2023. It just more reflects that we more shift the focus now on the data quality, because we finished with our general round of inspections. So what we inspect in terms of data quality? We of course look at the processes, responsibilities, and the adequate training of the involved personnel. We do request the line listings before the inspections of all the cases, post-marketing cases in our case, and our GCP inspectors look at the clinical cases. And you can see quite a lot already from the line listings usually. And then we choose a number of cases to review during the inspection. We often ask for a demonstration of the cases in the database and to see the processes there. And we review the full case documentation, and we are able then to check whether the data in the case is actually complete, versus the data that was reported, whether the information is coded correctly, assessed correctly, and so on. We'll look at the follow-up process and the documentation, including the targeted questionnaires, and so on. And of course, we also concentrate on data integrity and access control. And the common issues we see here are the findings in our inspection. Over-reporting is something that comes really often. There are the cases that are not valid, should not have been reported to the vigilance, but were reported by mistake. This could be the pregnancy cases, the off-label cases, death-only, drug intolerance, not related cases, and so on. The underreporting is also an issue that we see quite often. That's usually due to the fact that the companies missed some of the data that makes the case valid, and by mistake identified the case as non-valid, or they have a issue with their reporting rules that also comes up sometimes. Then we have the duplicate check that is ineffective. We have some issues with translations from native language to English, where the companies use automatic tools with no quality check. That can sometimes lead to underreporting as well. And then we have issues with the follow-up, also quite often with the overloaded, not really structured well follow-ups, where the reporters are just bombarded with a huge amount of questions and very lengthy questionnaires that actually doesn't add to anything to the case, but scares them off, and they do not provide any further information. And then we have quite often we detect issues with seriousness assessment. The cases that are received from third country or partners that are not reassessed and reevaluated. And again, seriousness assessment, because apparently I thought it's so important that I put it there twice. We also see quite a lot of coding issues in our inspections with the incorrect coding of events, the non-precise coding, the incorrect coding of the outcome, where we have seen cases with event death as recovered and resolved, or other issues of this kind, and other examples. So this is pretty much what I wanted to show today. Thank you very much for your attention. And
Yeah, thank you, Elena, for sharing with us some insights from how NCAs are processing their cases, but also sharing your interesting inspection findings with us. We are running a bit late, so I would like to move on to the veterinary NCA point of view by introducing our next speaker with Dr. Catherine Sherman. She's a veterinarian by training and joined the BVL Department of Veterinary Medicinal Products in 2017. And since then, her work in the Unit for Pharmacovigilance has mainly focused on adverse events and signal management at both national and EU level. And as a chair of the pilot Signal Management Expert Working Group, she's actively involved in the development of the signal management processes under the new veterinary legislation. And before I give the floor to you, I would also like to ask people, if you have any questions, please put them in the chat or during the Q&A that will follow after this. We may have a short time frame for that. Raise your hand. First, Catherine, the floor is yours. We cannot hear you yet.
Can you hear me now?
We can hear you, but I don't see any slides yet.
They should be there now. Can you see them?
We can see them. Yes. Now you're ready to go.
Perfect.
Thank you.
Thank you very much, and thanks for the introduction. So let me just jump right in since we are short on time already. So first, when I prepared for this presentation, the question one always asks themselves is, why is data quality important, and is it important for everybody, and why are we doing all this? So when you go through the internet, you find many of definitions as well as many reasons. However, I found this one on the IBM website, which I found quite fitting. So I'm just going to quickly read it. When data quality meets the standard for its intended use, data consumers can trust the data and leverage it to improve decision making, leading to the development of new business strategies or optimization of existing ones. So what I find most important here is meets the standards, and has already been highlighted by Anna in her presentation earlier today, that the standards are key, that we need to know what we want out of the data, and then we can set the criteria of what we want to see. So first we need data quality to know our standards and having them met, and then also we need them fit for purpose for the intended use, and ultimately we want to be able to trust the data. So I think this is quite important, because any conclusion, any possible regulatory action taken out of the data analysis after signal management, for example, is based on this data. So we need to be able that we have good data in and can trust the outcomes. So before I go into detail into what we do at the BVL side regarding data quality checks, let me just quickly recap, especially for those not so familiar with the veterinarian side, the reporting pathways. We have, as has already been mentioned in the presentation earlier at this session, a new veterinary legislation since at the end of January 2022. So in the old legislation, up until the end of January 2022, we had obviously the key stakeholders. We have a primary source or reporter, we have the national competent authorities, as well as marketing authorization holders, and we have the Eurovigilance veterinary system. So the source reporter can report to either a national competent authority or a marketing authorization holder. But back then, all reports received by a marketing authorization holder were submitted to the national competent authorities, and vice versa. National competent authorities submitted any case received by them to the marketing authorization holders. And ultimately, it was only the national competent authorities submitting reports to Eurovigilance Veterinary for the European side. So the NCAs were always the contact point or point of control for data quality, because every case would go through them. And as long as all competent authorities were harmonized and agreed upon certain standards among each other, we had a quite good feeling that data quality standards would be met and we had a control over them. So now with the new legislation, which has now been into place for a little over 2 years, we still have the beginning is the same. reporting to either national competent authorities or marketing authorization holders. But here we have a big change, that because now marketing authorization holders themselves report those adverse event reports directly to Eurovigilance Veterinary. So NCAs do no longer see every report going through. On top of that, marketing authorization holders do no longer receive directly those reports from the national competent authorities that were received by them, but have to retrieve those from the database themselves. And this is per legislation mandatory. If they're downloading it into their own databases, or if they're just taking note of it and having files, for example, Excel files where they keep track of them, is totally up to the individual marketing authorization holder. However, it's mandatory that they have knowledge of all reports regarding their products in the Eurovigilance veterinary database. This is not mandatory on the competent authority side. First, because not all competent authorities have own databases, and also because it is per legislation not explicitly expected that every case is being reviewed. On top of the new reporting pathways, we also have new reporting timelines. So while in the past, in the old legislation part, we had the possibility to not submit non-serious reports to the database. There was a 15-day reporting timeline for adverse events with the classification as serious. Marketing authorization holders back then had the possibility to keep non-serious reports at their end and just submit those with the periodic safety report updates. Since under the new legislation, we no longer have on the veterinarian side periodic safety report updates. We have now a 30-day reporting timeline, and every adverse event report received by either national competent authorities or marketing authorization holders has to be submitted within 30 days. So within those 30 days, we expect now kind of a very complete overview of the report of the adverse event to be submitted. And it has been also communicated that follow-ups should only be used if there's really important updates on the information within that for that specific adverse event. Because with the 30-day timeline and everything going directly into the database and duplicate checking and everything going on, it's quite important that the adverse event report is also pretty complete and in a good state once it's submitted for the first time. So let me just quickly talk also a bit about our national database. So the BVL is one of the national competent authorities on the veterinarian side that has their own database. And obviously, our database is, according to the legal requirements, capable of sending and receiving adverse events reports in the VICH standard, and it has passed the tests with the EVVET database. Both were mentioned in the presentation earlier by Laura. So having met those criteria, our database is in a position to receive via rerouting all of the German veterinarian reports reported to the Eurovigilance veterinary database. So whilst marketing authorization holders have to go into the system, look for the reports, retrieve them, we are in the luxury position that national competent authorities can have them just sent through the system. So once the system, the Eurovigilance veterinary database, receives a report, an adverse event report that is has occurred in Germany, it's automatically being submitted also to our database, rerouted. The reason we're having a national database is because it facilitates adverse event entry, review, and submission to EVVET. It also, of course, allows us a good overview of checking the receipt of acknowledgements, meaning that the case has passed through the through the gateway to the Eurovigilance database, to EVVET, and has been accepted. In case of failures, we check that also. Then this case is being adapted, changed, necessary changes are being made, and then resubmitted to EVVET. It also allows us an easier duplicate detection by assigning the reports to specific assessors, and then in this way we can see easier if a report, for example, has been submitted by a veterinarian to us as a national competent authority, as well as to the marketing authorization holder, and then we can liaise and discuss and take necessary action. It also facilitates surveillance on our end by being able to apply certain criteria of how we want to see our data in a day-to-day surveillance. And on top of that, it facilitates archiving of adverse event reports. And just to give you a quick idea of numbers of adverse events reports submitted in comparison to the human side, pre-new veterinary regulation, we have received roughly around 1,500 reports in Germany for the pharmaceuticals, I have to say that. And since the new veterinary regulation, we are approximately around 4,000. This increase is mainly due to the mandatory reporting to the database of non-serious reports. So what do we do on a day-to-day basis on our national competent authority side is that, and this is not an exhaustive list, so I just put there examples. So we definitely check each and every report. So every report that concerns a pharmaceutical veterinary medicinal product and has an adverse event in Germany is sent to us and checked by one of our colleagues. So what we definitely always do is we check the minimum criteria, obviously. So for example, is there only one species per report? Because the guidance states that if there is a report submitted by a reporter with, for example, a dog and a cat, this case has to be split into two, so we have one per species. We also look into the correct entry of the veterinary medicinal product name or active substance in the respective fields. The bedrock coding, according to description in the case narrative, is looked at. And it is also checked that there's no confidential information included in the case narrative. For example, the e-tag number of a cow or the name of an owner. All those kind of information needs to stay at the primary reporter's site. So it shouldn't go to the database and then to us. Within the case narrative, the focus is that all known relevant clinical and other related information is included in that case narrative. So anything that is in the data fields also should be stated in the case narrative. And this is per guidance as well. The VGBP guidance states that. So we check for that because sometimes there is a report where you see that certain data fields have been entered, however, the information is missing from the case narrative. This needs to be there. Also, the other way around, if it's possible, if there is a data field for something mentioned in the case narrative, this should also be provided in the data structure field. So this brings me right away to common errors and data quality issues that we come across. One thing that we have noticed is database compatibilities, incompatibilities, perhaps incorrect mapping, because some of the issues that we run across, we're not quite sure if this is an issue, if this situation is due to incorrect handling, incorrect entry, or perhaps incorrect mapping. Knowing that still a lot of conversions back and forth are going on, there might be also an issue with that. Even though all databases are being checked, we sometimes have, we're sometimes wondering why some situations arise. Data entry, as mentioned on the previous slide, we actually see that, for example, one report includes multiple species. What we also see, unfortunately, not quite that seldom, is the entry of an active substance in the brand name field, which leads to issues later on for the data analysis. Because if the active substance in a brand name field is mentioned, the coding and then the assigning of that report to the marketing authorization holder in EBBET for the analysis part becomes quite difficult. And in that situation, this report shows up for each and every marketing authorization holder with a veterinary medicinal product authorized with that active substance. So we keep communicating that, and so we're hoping that one day active substance will no longer be mentioned in the brand name field, but rather only in the active substance field. Also, if an active substance is known, ideally the use of unknown prior to the active substance should not be there. What we notice as well is wrong or non-coding of veterinary medicinal products from other marketing authorization holders. For example, incorrect spelling or not inquiring about the brand name. So we know that obviously a marketing authorization holder is most interested in their own products for data quality reasons and a better overview and analysis later on. It would be very important to have that information. So please, whenever possible, and you can find out the name of another VMP, even if it's from a competitor, please do so and enter it correctly. Some things like incorrect spelling could be easily handled by just doing a quick search, for example, online. What we also see is that the better coding in the respective fields is not matching the case narrative. For this, there's also specific guidance there. It's stated that it should be coded very closely to what the reporter has mentioned, and then we would expect that as well. Regarding the case narrative, we see, as mentioned before already, incomplete case narratives. So, for example, the age of a dog is coded in the field, but it's not mentioned in the case narrative. Or the other way around, the age is mentioned in the case narrative, but not entered in the data fields. And for some of the data fields, this is not mandatory to enter this. However, when we do an analysis and we have data fields available for that, it would be very helpful to have the respective information entered there to allow for an analysis on that field. Because otherwise the information is lost there, or it's becoming more difficult to find the data to be able to draw the right conclusions. What we also see is that a breed may be mentioned in the case narrative, but it's not coded. It could be that there is a Labrador dog concerned, and it's mentioned the breed Labrador in the case narrative, but when we look into the breed, it just states other, for example. So this is also something that we've come across. So what would be the actions taken after our day-to-day work and finding those potential issues? We can contact the marketing authorization holder, inform them and liaise with them on the issue detected, if applicable. It always depends on the situation of the issue that was detected. What would be the next step? Since we no longer can adjust or revise actively the adverse event report in the database, it would have to be the marketing authorization holder. So if there is something where it is viewed from our side that the case needs to be adapted, then definitely the marketing authorization holder would be contacted from our side with a question to please do that. On top of that, we have the entry in our internal list. So it's an internal way of record keeping of issues detected, which is obviously shared with the inspectors. So in that list, each and every issue is recorded and with the case number, which then also facilitates the work for the inspectors when an inspection is coming up. Because what we do is, not only do we do the review and data quality checks for the day-to-day work, for the analysis and for everything, but also to keep notes and track, to keep inspectors informed so that they know, for example, if perhaps an inspection needs to be triggered, or that they have a better overview of the situation regarding adverse event reporting and data quality when they go to an inspection that might be scheduled anyways. So for that inspection, we not only provide the review of reports in general, we also check timeliness of reporting, the coding and SOP review is being performed as well. And during an inspection, the marketing authorization holder is also asked to demonstrate their system, to show to the inspectors and, if applicable, assessors that might accompany the inspectors for that inspection, to show that how they deal with an adverse event report from the point of entry where they with the first contact with the source of information, all the way through to submission to EVVET. And even further now, since it's under the new veterinary legislation, a requirement for the marketing authorization holders also to show us their actions regarding signal management. So all of the information that we gather from checking the day-to-day case reports is also being shared with the inspectors. On top of the national actions regarding communication with inspectors, we also have now an EU list, and James Mount mentioned it quite briefly this morning, that we have the pharmacovigilance Working Party Veterinary now has a data quality subgroup, which was initiated to discuss data quality issues that have been noticed, to draft possible solutions and facilitate exchange to inspectors. Because this group, established in early 2022, I think it was, when we had the change of reporting pathways, has members coming from the pharmacovigilance assessor side as well as inspectors, to ensure that we have an exchange and that we have open discussions about possible issues. So basically what we have now is a collation of data quality issues within the EU and a list that allows us on a day-to-day work as well to report and let colleagues across the EU know of any potential problems and issues that we came across. Because even though we may notice a reporting issue with one marketing authorization holder, it may not necessarily be that that marketing authorization holder is being inspected by our agency. It could be that the PSMF is in a different member state, and so a different member state authority would be responsible. And this exchange at EU level allows us to easier have a more frequent exchange on top of the already in place structures, where usually before an inspection there is always the question to other member states. So just briefly before I'm finished, the use of adverse event data. So what do we use the data for on national level? It's for reporting, for example, our yearly statistics that we publish. It's for signal detection, analysis, and general surveillance. And here it needs to be highlighted that even though we have the day-to-day check of all reports coming in, during signal detection at national and as well as at EU level, we do perform another check again. Because when we go through each and every report for a specific product, obviously we would catch on if there's something systematically a problem, for example. On top of that, here at this, we also noticed that an issue that's coming up now, but it's not really new, is recoding. That's something that we came across, is that, for example, in the past, it was prior to PSUR being submitted that sometimes there would be a review for a specific product of the reports, and then all of a sudden we would receive plenty of follow-ups because the federal coding would be changed. So we had and have that still occasionally, that all of a sudden for a specific product, the terms are changed. And then this obviously changes also the analysis in the database. And so this is also something that is coming up now, and where we need to identify how to deal with that data quality issue, because with a 30-day timeline for reporting, we would expect that a certain level of quality check has been done, has been performed prior to submission, and it should not necessarily require later on the adapting of each and every report with a certain specific better term to be changed to another term. And this is something that is coming up again and will be discussed in the near future. On top of that, we obviously use adverse event data for publication of data for specific topics that we came across when we queried the database, so to keep the general public or the specific public, like veterinarians, informed. And with that, I thank you for your attention, and thank you.
Thank you very much, Catherine, for this interesting presentation. It's also interesting to learn during this session that for veterinary and human side, the basic processes to improve data quality are quite similar. We also run into similar issues identified when it comes to coding or structuring data. And Catherine, I have to say, I'm impressed with the effort that the German Veterinary Agency is putting into addressing data quality issues really at individual case level, and that it's embedded in your day-to-day process. I'm really impressed with that. We are running over time. We had allocated originally some time for questions and answers, but I have to say I do not see any hand raised, and I was monitoring the chat, but there's nothing in the chat. So before we move to a next session, I just wanted to ask a question myself to Elena to clarify one thing in your presentation, because you also used the word underreporting. And I think, Elena, in the context where you used underreporting, it has to be interpreted as an MAH has received ADR info and is not submitting this as an ICSR to ultravigilance. Where I've also used the word underreporting in my presentation, but there it is to be understood as primary sources are not informing NCAs or marketing authorization holders of their suspected ADR.
Yes.
Yeah.
Sorry.
Okay.
Yes, in our case, it's obviously the cases that we identified during the inspection that do not find their way in ultravigilance at all, due to the fact that the marketing authorization holder, by mistake, identified it as a not valid case or didn't see the information. Yes.
Yeah. Okay. And now there is a question in the chat, but this is about it's for Elke about the clinical trial. And I think we have Elke in our next session. So if you're okay, I will refer it to the next session and close session 2 and move to session 3. Elke, can I give the floor to you as the chair of session 3?
Yeah, thanks, Anja, for handing over. I think we'll take care on the question in the chat after the presentations. I think we should have some time for it. Well, session 3 now is dealing actually with stakeholders' way of ensuring quality in ADR data. And it's a pleasure for me to introduce you Jim Neckes. He will present what's done in the Human Marketing Association holder world. Jim is currently the Group Vice President and Global Head of Pharmacovigilance and Clinical Medical Writing at BioMarine in the Bay Area in California. He joined the company in 2010 with more than 12 years of academic experience at UCLA Medical Center and 18 years at GeneTech. So, floor is yours.
Thank you. Can you all hear me?
Yep.
Are you going to project my slides, or do I need to share screen? I was told that you had them.
Oh.
Yeah, I can share for you. Just 1 second, please.
Well, while the slides are coming up, I'd like to thank the EMA and the European Confederation of Pharmaceutical Entrepreneurs for the opportunity to participate in the workshop on data quality framework for ADRs. I'll be presenting marketing authorization holder perspectives on this very important topic. So happy to see the focus on this, and we've all heard the adage, garbage in is garbage out, and it's so true. And in this area, to really do good safety surveillance, we really need, good quality data. So next slide, please. So with respect to ADR quality activities in Europe and elsewhere, actually, not just in Europe, there are several actors, involved who can influence data quality or provide analysis of data. First and foremost, we have patients, who are direct reporters of safety information through either spontaneous reporting systems or patient support programs that companies run. And they're responsible for, for accurately describing their symptoms, medical history, and medical use, medicine use. We've also heard from regulatory authorities already how you all maintain ADR reporting databases, you receive and process ADR reports from healthcare professionals and other sources. You perform the data quality checks and coding and signal detection on an ongoing basis. You also assess the benefit risk of our products on an ongoing basis and take action when necessary. And very importantly, which you're continue to evolve, is to providing guidelines and standards for ADR reporting and data quality, which is extremely important. We have the healthcare professionals, obviously, physicians, pharmacists, nurses, who are primary reporters of ADR data through spontaneous reporting systems. And very important, they're responsible for accurate and complete documentation of patient medical histories, diagnosis, and treatments, which doesn't always happen to our liking, but that is an important aspect of this. They also can help provide clinical context and causality assessments for reported adverse drug reactions. We have researchers and pharmacoepidemiologists who often participate, that companies often recruit to help analyze ADR data from various sources, including spontaneous reports or electronic health records and clinical trials. And they also help us develop and apply methodologies sometimes to assess the quality and identify bias and improve our detection capabilities. Importantly, especially in the electronic health records situation, if we aspire to use those data to find safety issues, the medical coders who code the diagnosis, the procedures, and adverse events are important. They can ensure the accurate and consistent coding practices to support the analysis that we're all trying to do. We often engage drug safety committees who are comprised of experts, patient representatives, or regulatory authorities to help review and evaluate our safety data that we collect, either through clinical trials or from other sources. And they oftentimes, especially in the clinical trial setting, provide recommendations on risk management strategies and regulatory decisions. And last but not least, pharmaceutical companies. We maintain pharmacovigilance departments and drug safety systems. That's our expertise. We collect and process ADR reports from all the sources. We conduct, similar to the health authorities, data quality checks. We perform follow-up investigations, causality assessments, which can be challenging. We analyze the safety data on an ongoing basis to identify potential signals. And we're responsible for reporting the signals to the regulatory authorities and other stakeholders. Importantly on this slide is that collaboration and effective communication among these various stakeholders is essential for ensuring the quality of ADR data and supporting our decision-making with regard to drug safety. Next slide. There are a number of things that are already happening, to improve ADR reporting and data quality. From the manufacturer's point of view or marketing authorization's point of view, where we establish robust pharmacovigilance systems and processes for ADR collection, processing, and reporting, similar following the guidance that is provided to us by the health authorities, which incidentally, EMA's guidance is very robust and excellent, but we don't have harmonization all the time across the world, which makes things challenging. We're also involved in training healthcare professionals and patient support programs on the ADR reporting. That's an ongoing effort. We implement quality control measures such as data validation, duplicate detection, and causality assessments similar to the health authorities. We spend a lot of effort conducting follow-up investigations and collecting additional information to enhance data completeness. That can be challenging, not always successful. We're involved in analyzing the ADR data to identify potential signals and trends on an ongoing basis. Collaborating with regulatory authorities and healthcare providers throughout the life cycle of products is important. We've had many discussions, at least in our company, and I'm sure others, on how to improve the reporting during programs that are conducted. And we develop and implement risk management plans to mitigate, potential safety issues. Again, closing on this slide, there's continuous improvement efforts and collaboration among stakeholders that is necessary to keep this improvement going. There's new technologies that are emerging that can help artificial intelligence, natural language processing, and these things can further enhance data quality across different stakeholder groups. So really pleased to see this effort to really bring the groups together, solicit input to try to develop guidances to really hone in on what can improve the quality, because at the end of the day, that's going to be instrumental in helping us identify new safety issues, communicating them, and to optimize the benefit risk of the products. On the challenge, there are challenges in measuring, characterizing, and improving data quality. Some of them have already been touched upon from other speakers. There's the underreporting and of ADRs can be a problem. Sometimes only a fraction of events get reported, leading to incomplete picture of the product's profile. There can be reporting bias sometimes, where there's a tendency to report maybe certain types of events, serious or for novel ADRs, focusing on those. That can skew data and make it difficult to accurately estimate the incidence sometimes. Measuring the extent of underreporting and quantifying reporting bias is challenging. Sometimes the true denominator or the total number of ADRs is hard to estimate with our current systems. There can be incomplete, this was discussed already quite a bit, incomplete and inconsistent data. ADRs reports often lack important details, such as patient demographics, concomitant meds, medical history. You would think these things are pretty basic, but they don't always come in the way that we want them. And in fact, this might be part of the effort to try to really communicate what makes a great report or a complete report. There are inconsistencies in data capture, coding practices, and terminologies across different systems that could hamper data integration and analysis at times. Measuring the completeness and consistency of data queries and review and validation, which can be resource intensive, is an issue. Data quality can vary across sources. All right, ADR data quality, such as spontaneous reports and electronic health records and clinical trials, they all have their strengths and limitations. and combining from multiple resources can be challenging because the normalization doesn't exist across these different sources sometimes. Characterizing and accounting for varying data quality across sources is crucial for accurate signal detection. On the signal detection and data mining challenges, identifying true safety signals amidst the noise and uncertainties in ADR data is a complex task. We've heard about the noise situation. I think that's an area of focus. Actually defining, what is suspect. There's still quite a bit of noise that gets reported, for example, incidental events, for example, that may not have been the intent of the reporting. There's still quite a bit of effort involved in processing that information, and sometimes it gets reported and it might, cloud databases with things that are probably not drug related. There's factors such as data sparsity, missing data, confounding variables, and temporal relations between drug exposure and adverse events can complicate signal detection efforts. Measuring the performance of signal detection algorithms and validating their findings is difficult due to the lack of comprehensive ground truth or ADR data. And then lastly, there's continuous evolution of data standards and practices, and they keeping up with them and harmonizing them can be challenging sometimes. Implementing and adapting to changing standards and practices across different stakeholders and systems can be challenging at times, particularly in terms of ensuring data consistency and comparability over time. Measuring the impact of changes in data quality practices and quantifying improvements can be difficult, as historical data may not directly be comparable to current data. So addressing these challenges requires, I think, some collaborative efforts among all the stakeholders that are participating today. Strategies such as harmonizing data standards, implementing quality control processes, integrating data from multiple sources, applying advanced analytical techniques, and leveraging emerging technologies such as AI can help improve the measurement, characterization, and overall quality of ADR data. And quite a bit of progress is being made already. And again, pleased to see the focus on data quality in this effort that's being discussed today. Next slide. We've talked about this already, but let me just focus a little bit. This will be my last slide, I believe. There's different ADR reporting routes, and they're all associated with certain challenges. For spontaneous reporting systems, some of the challenges that have been highlighted already are underreporting can occur. Only a small fraction of ADRs get reported, or sometimes what gets reported is probably not an ADR. Again, it could be incidental medical events, which CIOMS defined quite some years ago. And we still, as I said earlier, spend a lot of time processing that kind of information. And in some parts of the world, we're required to still report it. I'm glad that we, in the EMA, we have the ability to kind of weed that out sometimes through the solicited aspect of reporting. However, sometimes that's not easily done because we're lacking information. So noise can get reported. There can be reporting biases, factors such as severity of the ADR, the novelty of the drug, media attention, things like that can influence reporting rates. We've already talked about incomplete information. Reports may lack essential details, demographics, ConMeds, clinical context, making causality assessments difficult. Already talked about duplicate reporting. Multiple reports of the same ADR can lead to redundancy and skew risk estimates. Patient support programs, which are used extensively in industry to help, educate patients and optimize use of the products. But there are challenges with regard to data quality. Patient self-reporting, patients sometimes lack medical knowledge, which can lead to misinterpretation of symptoms and inaccurate reporting. Now, they're an important source. They should know their body, and in fact, focusing in on what's different, what's changed, is really the counsel that should go to patients. But nonetheless, there can be sometimes reporting from patients that is hard to sift through. Over-reporting, patients may attribute adverse events to the medication, leading to false positives, especially incidental events that were not the intent of what they wanted to talk about. They may just be mentioning their medical history. So sometimes that information is collected and processed anyway. Patients may not provide detailed clinical information necessary for causality assessment. On the electronic health record medical claim database front, some of the challenges there are coding inaccuracies, incorrect coding, or the use of non-specific codes can lead to misclassification of adverse drug reactions. Missing data, we've talked about that already. Relevant clinical details may not be captured in structured EHR fields or claims data, limiting the ability to assess causality for that. Confounding factors can be challenging to establish the causality link between a medication and an adverse event due to the presence of, let's say, comorbidities or con meds or other confounding factors. And lastly, on the clinical trial and post-marketing study front, some of the challenges there that have been touched upon already also, generalizability. Clinical trial populations may not be representative of the real-world patient populations, which limits the applicability of the findings sometimes. There can be limited follow-up as well in some of these studies. Post-marketing studies often have limited capture long-term or delayed adverse drug reactions, for example. And there can be reporting biases. As with spontaneous reporting systems, reporting biases can exist, leading to incomplete picture of ADR profiles. So maybe that got us back on time. I speak fast, but those were my remarks, and I don't have any other information to present.
Thanks, Jim. I think for the matter of time, we move to the next speaker and see if you have questions, please show up your hand or write your question in the chat, please, for the Q&A session afterwards. Moving to quality assurance in the CRO setting, and I would welcome Surab Korbadise. He is the Director of Medical Safety at FGK Clinical Research and member of Pharmacovigilance Working Group at the CRO Federation, the European. And before he joined FGK 13 years ago, he was working and also producing his own investigator-initiated clinical trials and worked for medical aid and charity in developing countries. So I would hand over. I'm not seeing you currently.
Can you hear me?
Yes.
All right, so it's already first.
Okay.
And the video is there, and the presentation should be coming.
Not yet.
Now?
Not at my side, to be honest. Maybe try again to share.
Yep. Share. This. Share.
Coming. Okay. Fine.
Well, thank you very much for this opportunity to speak about that very important item that keeps us busy in the safety groups in the CROs. And thank you to the previous speaker that he saved some time, so I will use this time, I promise. And so let us start. And the first we're gonna be in the coming 15 minutes. We will take just a quick look on the place that CROs take in clinical development and their work in medical safety, in drug safety, that would explain the role CROs play and will play in enhancing of quality of ADR data. Then we will outline the main steps and actors involved in ADR management in clinical trials. And we also touch base on the challenges that on each step, in each step of the safety information flow is. Then we will be talking about the current activities that CROs undertake to improve the data quality, the safety data quality in clinical trials. And also potentially, if we have time, we very quickly will look at potential developments that we expect to be implemented in clinical trials from CRO perspective to improve the quality of the ADR data. So let us take a look at first on the role of CROs that CROs are playing in clinical development. One has to admit that over the course of the last years, the CROs, and particularly big and global CROs, are taking ever increasing role in pharmacovigilance work and working with the marketed drugs, and such are also influencing the spontaneous ICSR reporting. However, the focus of my presentation today is something that is traditional CRO business domain, which is the clinical trials and solicited reports that we collect. Now, CROs, we all know, do help pharma undertake many elements of clinical trials, and that without the need for in-house facilities and bringing expertise in regulatory affairs, bringing global presence, and bringing expertise in the quality with them. And this is specifically important for small biotech companies that consist of these all bright minds that come together, develop their products, and bring it to the clinical development, but then they lack experience to push through the clinical development. That's where the CROs play very important role. And according to the survey that was conducted in 2023, CROs are estimated to receive, to be outsourced for 55% of the clinical trials conducted. And actually, the survey also has a data that refers to the years before. And when we take into consideration the years before, it turns out in 23, the percentage went a bit down. So on average, it's around 60% of clinical trials are conducted by the CROs. And when we take a look at which size pharma companies are outsourcing and what type of clinical trials, that not surprisingly, we see that the small biotech companies are taking the lead here from 60 to 76%. They do outsource clinical trials to CROs. So, in addition to that, we estimate ourselves that the majority of clinical trials conducted in general by the nature of the clinical trials are conducted, started and financed by the small biotech companies. And that's why the CROs play an extremely important role in the clinical development. Now, in addition to the classical offerings of the CRO business model, which, as we know, includes, for example, things like project management, regulatory, or statistics, or medical writing, most medium, and I would say all big size CROs do have medical safety departments. And within the medical safety departments offering, they do have a package of the core medical services that they offer. And that includes clinical monitoring, case management, databasing, regulatory submission, periodic benefit risk assessment. In addition, there is also this additional package that mostly, as we said, big and global CROs are now moving towards, or already moved into that direction. It's a package of pharmacovigilance services and the package of specialized for non-interventional studies, for example. Just an examples, brief examples. Now, as a result, we can estimate that in majority of clinical trials, CROs are actively managing safety data. Now, clinical development is a highly regulated environment, right? So in this environment, it's all described who is doing what. So you name it, every role is described and responsibilities are described. And if it's not described in the legislation, then it should be described in the protocol. However, our experience shows that despite, or maybe because this is all well defined, then the CROs are still addressing or implementing or employing quality measures on each step that is presented here to improve the quality of ADR data. I split the safety data management process in this presentation in 4 steps, just to represent the potential targets for the quality improvement that the CROs are employing. So we start with the study site. So the study site is the place where at point of care, the information from the patient is collected, all the clinical signs, and then it is recorded, right? So traditionally paper, now it's more electronic, and then there comes EDC, electronic data capture system. So electronic data capture system is a vehicle or vector that is used by the study site to put data in, and then through this vector that information goes stored and then goes to the safety group. Now the safety group looks at the data, does analysis, and transfers it further where it's needed. And there is also important step of submissions. Now with the submissions, we do have different types of submissions, and it's also important to make sure that there is a quality of data that you submit. So you might have a wonderful case in your database, but it does not go through to the UDRA vigilance. So you need to be aware. Now, in addition to these steps, within each step, actually, there are some challenges. And this is just not complete list, obviously, just a few examples what the challenges are. For the study side, you need to be aware, and the CRO has to take into account when the study is to be designed or conducted, that there is a variability in experience and training level. If we are talking about the electronic data capture system, then there is this traditional clinical database that includes adverse events, and there is a safety database that includes serious adverse events. And then there is a question, how up to date are two databases at any given point of time, and this is not the case always because of the nature of information, how it goes in. There is also a question on the data security of the data that is stored in the electronic data capture system. Now, with the safety group, there is always, if you talk about the trial complexity, influences the work of the safety group. So you can have within one clinical trial, several CROs working together. How do you coordinate with them? If you have big global clinical development program, then how different CROs coordinate with each other. And there is a question on the regulatory changes, how quickly we adapt to them as a challenge. And then there is always question on the staff recruitment and retention of the talents. On the current market, it's a very tough thing. And in terms of submission, we do in clinical development have different types of submission of the different types of safety data. It could be expedited, periodic, and then it could be safety data that comes for the purposes of the clinical study report or for the purposes of preparation of the submission dossier. Now, within these current steps, obviously, these steps are not something new, and that's why CROs are already equipped. They know that it needs to be addressed. And the best practice is, of course, to start before the study starts. And that's why it starts with the providing clear definition of safety terminology in the protocol. So the safety group should be part of the protocol development for sure. Now, there are also specific documents that further describe the safety specifics for the clinical trial, so-called safety management plans. That's where we are trying to improve quality of collected adverse drug reactions in the future by designing SIE collection forms in such a way that they are easily understandable for the sites, that they're easy to fill out, they don't bring a lot of burden, and they're study specific. So they are talking about things that are required for the protocol. So we are not collecting something that is not needed potentially. And then there is also the need for continuous training and sensitization of the study site, and especially PI, starting from the study site initiation and during conduct of the study. And this sort of training focus can be, of course, what we all know is the timely reporting of SIEs, right? So the 24 hours, or you can just an example of emphasizing that the diagnosis should be reported versus symptoms, or the final diagnosis when you know this should be reported, and then usage of, for example, I don't know, a CTCIE system for severity assessment, of the protocol defined system of severity assessment and grading, and other examples. So that kind of training. And then there is a question that is still open, is the quality of the narrative that comes from the site, right? You can do your best to try to train the site, but still it's an open question what comes from the site because of the various objective and subjective reasons. And the good thing is to have a medical monitor on the side of CRO to work with the site and make sure that the narrative is improved. And then this, what I mentioned above, are pretty much like a preventive actions that you need to take into account. There is also one corrective action that you can implement during the conduct of the trial. You can monitor the clusters of incomplete reports coming from the sites. And if you observe that these incomplete reports are coming from specific site, then you can address this. So this kind of quality measure to make sure that the site is reporting. So you need to find out the reason why there is this bad reporting coming. So this monitoring of the cluster monitoring. Now, in terms of the electronic data capture system, what we implement is implementation of automatic checks within the system. So if there are obvious inconsistencies in the data entry, the patient is 200 years old, or I don't know, some other parameters, right? Then the EDC system is automatically highlights it for the PI and asks, are you sure this? Please reconfirm. Okay, so the PI, of course, has the right to write 200 years if that's sort of desired, but nevertheless, the question is asked. So making sure that the quality is good. So there is no mistake on that stage. Then we are requesting signatures and approvals for important data entry steps, specifically for ADR reporting or the serious adverse event reporting, making sure that the responsible people can enter and confirm the correctness of the data. Now, and of course, considering that, again, there is a safety database, clinical database, etc., then there is a need for reconciliation. From the safety group perspective, when we are talking about the data entry, then of course comes into the question the principle of the 4I entry, right, as a minimum standard. Then safety group use, of course, standardized terminologies, either MEDRA or the WHO system for the coding of adverse events, medical history, concomitant medications. Then we are employing when we generate the cases for submission, we used templates, and this has, of course, a downside and as well as a positive, but nevertheless, it improves the quality for the poor reports, and one cannot underestimate the staff training, because we are the CRO is a human business, and there are many people involved, and they need to be highly specialized in what they do, and that's why the staff training is important. And again, because of this as a human business, we need to have, that is prone sometimes for mistakes, we need to have a robust company deviation management process that is open to intake all deviations, analyze them, and produce preventive and corrective actions that are accepted by the company. So very good deviation management is the key. Company deviation management is the key to improve the processes of ADR specifically. And then comes the submission, and just examples of what can be done, what is done by the CROs. It's of course the 4I principle when you're using, for example, the web trader submission. It's good to move to the submission that avoids this sort of need for 4I principle, like if you are employing gateway solution, but nevertheless, even gateway solution should not be relied completely, because if you don't get the acknowledgement of receipt, positive message, then there is some problem, and then the technology has to be reviewed. What caused this problem? So these are the current activities to improve the data quality in clinical trials. As I put together some examples, specifically, I would say related to the topic of today's workshop. Now, now talking about routine quality measures implemented by CROs, we should potentially also look at potential solutions that should be coming, that would be helping us further to enhance the quality of ADRs. And I have to admit that this future picture that you have on your screen might be, and most likely is already in place. So it's not like a future for some companies. I would say mostly big companies, because this implementation of artificial intelligence and all these beautiful things, it requires the implementation, it's a very time intensive and resource intensive exercise. So you need to have a lot of resources. So what happens is that it could be that the pieces of information that we receive is that the big CROs do potentially already employ some of this. But the small CROs are also grouping together to share resources and developing their sort of understanding and approach to the new technology that came in. What we see currently and potentially can be the target for improvement is the disconnect between some of the interfaces in the safety information flow in clinical trials. So, for example, we think that future will be in if we are bridging the data collection from the point of care to the storage of information. Okay, we all know about this, the wearables devices and everything, right? That record the data directly on the patient and transfer it. So this is exactly the thing that should be helping to collect data from the point of care directly to the database. However, one should also improve and include in this technology the quality methodology. For example, you can have a feedback mechanism to the wearable device. So if wearable device is identifying tachycardia in the patient and is reporting this to the central storage, then the artificial intelligence gets this information and sends back the question to the patient. Can you please confirm that you are not running or you are scared or something that can be objectively causing tachycardia? Just an example from our brainstorming of the situation. And then that should be improving potentially the quality of data that we collect in the future. Now, and obviously this flow of information should be going through with the help of artificial intelligence, but don't forget that we have on the side the human operators who should be controlling this, and also the quality control of the whole process by different methodology that still needs to be potentially developed. And but thinking about this, I would say that, and also about the topic of today's workshop, I would say that the future goal for us is, from clinical trials as a minimum, is not only to assure the highest quality of collected adverse drug reactions, but also collection of true set of adverse reaction and physiological signs from the patient as in response to the IMP. And thinking about the future of adverse drug reaction reporting quality, I discovered for myself that, I mean, a few years ago, we were looking for technology that would sort of address our needs. And now we need to keep up with the technology advances. And then, as I already said before, that what for some is perceived as the future, for others is already in the past. And this is the reality of different sizes of the companies and the different resources that they have. And the last point, and I think is very important, is that relying on technology alone is not the future solution. You need to invest in people who will be working with this technology, and you need in line enhance the quality methodology. Thank you.
Thank you. Thanks for your elaborated display of CRO and your thoughts on quality and improvement of quality and already performing quality checks. Matter of time, moving on directly to Magali Quentin, who gives us an overview on or insight on what happened on the vet side. Magali is head of Pharmacovigilance Europe and QPPV at Boehringer Ingelheim, Vetmedica, and she worked as a veterinarian surgeon in equine practice and moved to industry with diverse positions at pharmacovigilance in human and veterinary companies. So floor is yours, thanks.
Thank you, Elke. And thank you very much to, I'm sharing, but I cannot see.
You're sharing your slide, so that's working.
You can see the slide. Okay, then it's all fine. Yeah, thank you very much for the organizer of this workshop, and I would like also to express the appreciation of the veterinary industry to have the opportunity to participate. And today I have the pleasure to present the view of this industry on behalf of Animal Health for Europe, Access VetMed, and the Association of Veterinary Consultants. Working myself for a human company in the past, I thought it's appropriate to set rapidly the scene and point out some specificities of the adverse event collected for veterinary medicinal product. So quickly, we are facing multiple species in our report with different requirements for reporting. For example, farm animals may be considered as a group. An extreme case or an extreme example are the bees, which are treated as a beehive. For pet, it's usually one animal per case, but it's not always the case or the fact. We have also to deal with report of human exposure to our medicinal product, if it's symptomatic or not. For adverse event that we collect, we are collecting also very special type of adverse event, like incidents to the environment or violation of approved maximal residue limit, which bring with them different requirements for the setup of the data. And also you need to understand that mainly the business will drive the reporting of an adverse event, like a decrease in production performances or an increase in expected mortality. The reporter, the source is probably as various as in what have been described by my past presenter. We have mainly, or the huge majority of our report are spontaneous. It's a very little pre-marketing clinical trial experience in the database. And remember as well, pharmacovigilance for vet product start with the launch of the product on the market or with the marketing authorization. So no first-in-man, no SUSARs. As I said, the reporting is also very much triggered by customer care activities. Reporters are mainly animal owner, but also vet nurse from a vet practice or animal health technician on big farms. And this has also an impact on the case quality. The veterinarians are difficult to get on the phone again and again for follow-up, because they have limited time. They need to ensure their business. They don't get any payment via a health insurance system. We are also facing social media cases and with the same issue, probably the difficulty to identify the reporter in such cases, and the fact that the reporting is triggered rather by emotion than by medicinal facts. In terms of product, we report in a case at the brand level rather than at the substance level. So it is definitely a challenge to standardize rule for quality when you have such a diversity of reports. We need to take into account that there are some commercial elements that triggers the reporting for veterinary medicines, and the use of machine learning or artificial intelligence, had it is already practiced in human health, for example, from a form directly to a case in a database, does certainly not have the same parallel in animal health yet. You have already heard by James and also by Catherine, we have a new regulation in place since January 2022. The focus is now on risk management and on signal detection to ensure this risk management of our products. We have common tool that all stakeholders in the network are using. This is the Udra Vigilance Veterinary Database, the repository for the adverse event report, and the data warehouse, which is the tool used for the statistical analysis. This is based on the calculation of report odds ratio. And we don't write PSUR anymore, at least not in Europe. Outside of Europe, the picture is slightly different. I would like to say some words about the data model within ultravigilance VET. Apologize.
Is er ook een boterhammetje voor mij niet?
Just go ahead, please. Others mute.
Oh, sorry. So with regard to the VET model, we are reporting since January every single case that we collect, domestic or foreign, serious or not, spontaneous or solicited. Everything goes into ultravigilance VET. There are fields that are not mandatory anymore. We don't code anymore for any expectedness according to a label. We also are not expected to provide a causality assessment, but this is a practice that has been kept, to my knowledge, by most of the marketing authorization holders still. There is also no distinction in our report anymore between the products that have been used concomitantly or that are suspected to have caused the event that triggers a report. There are also some limitations on how we can report products that have been used for diagnostic reason. And if you now consider reports which concern multiple products from multiple MAH, and a very standard one is the first visit to the vet with the new puppy, vaccination plus an antiparasitics from two different companies. So the MAH, that's what I want to say with this example. As Catherine has mentioned, as MAH, we are expected to download all cases that have been submitted by other MAH when there is one of our product which is concerned. And we have a second rule that goes together with that on how we work in EVVET is only the reporter of the case, the originator of the case, can submit follow-up. If I have a product concern in the case, but I'm not the originator, I can only view the case, download it, but I cannot send follow-up or modify any information in that case. What does it mean? We have to exchange outside of the database, for example, via email, additional information that we may have received to a particular case. That causes definitely weakness to the data quality construct in EVVET. When we look into VEDRA coding, yes, this is a key point and a key focus now for the data quality of the cases because of the intended signal detection afterwards. And signal detection is based on a pair product VEDRA term. So if the VEDRA term is not present, you will not retrieve the in your signal detection activity. Another aspect, and it has been roughly mentioned, I think, by Laura and also by Catherine, is the recoding or the reclassification of product in the background. Here again, cases with multiple products from multiple MAH, the originator is not fully aware of the portfolio of the competitors, provide a name, and in the background it is recoded, reclassified in a way that the other MAH can download their case. This is also here a weakness because of wrong assignment of the product name by the system, or we know that third country products are still a challenge for the system. And we appreciate that all is a matter of improvement and that a lot of work is done in the background for that. We have identified certain number of challenges with the way we deal with the case now with the new EVVET. And the biggest one is definitely the detection and the handling of duplicate. And here all stakeholders are impacted because we are all working in the data warehouse, extracting data from ultravigilance vet. The current process, as we see it at the MAH level, is a process outside of the database. It's a one-to-one communication between one case owner and the duplicate case owner to decide on which case is maintained, which case get nullified, and potentially exchange additional information to complete the case that will remain in the database. And this is a huge workload. And this is the same when competent authorities the MAH to the idea is that the decision for the nullification is made based on the date when the case was first reported to ultravigilance vet, means there are no data quality aspects considered in that decision. That is also something that we want to look into. As international MAH, we face different requirements from different agencies. It regards VEDRA coding practice, it regards follow-up practice, it regards reporting practice. Europe is the only region where we can take up to 30 days to send a report. Most of the rest of the world remain with the oldest or previous rule, 15 days for a serious report. As long as this difference remain, we will continue, of course, to process our case with the highest speed to meet the more constraintful requirement we have in the world. And I would like also at this place say that with the new way of reporting everything, as I said, domestic, but also foreign into EVVET, the balance between case originating from EU and case originating outside of the EU is, to my experience within Boehringer Ingelheim Vet Medica, 80% outside of EU, 20% within EU, with the US as the absolute top driver for cases that we receive. So, of course, this balance brings me to look much more at what is requested in the US from a case quality and reporting timelines point of view. If we continue also to look at these cases or report with multiple product, multiple MAH, the VEDRA coding is also here an issue because it will not fit for all product, and we code at case level, not at product level. I give you an example. You have a dog on painkiller. He experienced pain after a vaccination. The MAH from the painkiller will code lack of efficacy. But the MAH from the vaccine does not want to see this lack of efficacy, because that you have pain after a vaccine is not lack of efficacy, it's probably a local injection, a local reaction to the injection. Another example, your animal, your cat is on long treatment for renal insufficiency. These are drops that you are giving every day. He shows one-time vomiting. 2 days later, he gets vaccinated and he developed a local injection site reaction. The MAH from the vaccine will code this injection site reaction. But of course, the MAH from the renal product, the drops, he wants to see the vomiting coded in addition. So this leads either to a case which is improperly coded for one of the MAH anyway, and then also for the NCA looking at this case. And it leads also in many situations to the decision to split the case, which is rather a discouraged practice because it prevents you then to look properly into where do I have interaction between two products causing adverse event. So with this kind of practice, I have the impression that we are moving away from the concept of what is a spontaneous report and what is the intent of the report. And it definitely impacts the quality. Okay. So in conclusion, pharmacovigilance practice, please, need to be adapted to the vet business, just because of this specificity I've mentioned at the beginning. Our PV teams are very much smaller than in the pharmaceutical industry for human medicine. My experience inside of BI or inside of Boehringer is that we have a ratio from 1 to 6 to 1 to 10. So for 6 colleagues working in pharmacovigilance, on the human pharma side, I have 1 person in my own PV team. And I would like to emphasize, and this will be my last message, something that we have already said at several opportunities and within several discussions with our authorities. From an industry point of view, we really see the need of an harmonization on the ICH level required in terms of data quality. Thank you very much for listening to my presentation today.
Thanks, Magali. Thanks for this interesting insight in the animal health situation. And we're coming to the last speaker of the session. I'm look forward to the quality addressing at a non-commercial sponsor. I'm inviting now Tula Ikonen from EOTC to her speech. She's currently working as a head of pharmacovigilance department at EOTC, and before she had several other positions in pharmacovigilance in pharmaceutical industries. So Tula, please.
Thank you very much, Elke, for introducing, and thank you for inviting me. I am sharing the presentation.
Yeah, fine.
Thank you. I think you see it now. So the ADR data quality activities of a non-commercial sponsor is my topic, and I took the liberty to put there a subtitle of safety data quality activities at AOTC. At first, I would like to say a couple of words about the landscape of non-commercial sponsors. A non-commercial sponsor is not an organization with one single profile. We have differences, for example, in our operational mode. You can have a national level non-commercial sponsor with one or few investigators having one or couple of centers, sites, or it can come up to a multi-center international organization with a quite large network of investigators with a centralized operational mode like units to handle safety reporting and data management. From protocol side, of course, the science is covered fully, and we have anything from single to multi-arm protocols. All phases are definitely covered, complex trials to pragmatic trials. Size of the trial usually varies per academic sponsor, and we have all type of data collection approaches. We collect indeed full safety data, or there might be a low intervention design or another risk proportionate approach to the data collection. We are not indeed a marketing authorization holder. We do not commercially own medicinal products. And what is also common to every academic sponsor is that we probably have multiple sources of funding. And AOTC positions itself here. We are indeed internationally operating a multi-center organization with a lot of investigators in our network. So what are our clinical trial safety data activities at AOTC? I focus here mainly on clinical trials. So this is a quite pragmatic point of view now. So of course we look into our data, which is individual case safety reports, including SARs, SAEs, SARs, and SUSARs, and also AE data. It's here because sometimes for a non-serious AE data, it is spotted that actually that might fulfill the criteria of seriousness, which changes the classification of the data to a serious report. Then we have, of course, aggregate reports, DSURs, meaning nowadays annual safety reports. We look into the timeliness of the data at the receipt of the data, and also when we report the data further, submission compliance. For database, we check the correct functioning of the databases. For example, we do database testing and validation per project. Here I roughly divided our data quality activities timely in two phases. Before data collection, meaning before the trial can be activated, there we start with a database setup where we always routinely review our AE and SA CRFs. We do database validating and testing. And as a little example of specificities, if we have specific events which are to be collected with a specific time frame, we need to take this into consideration in this phase. At the data collection, we have several AE reviews for serious data. We review essay queries. We do a quality check for ASRs and TMF. And I took also here the source data verification that is performed at monitoring visits. At us, the reviews are performed by our data management team, pharmacovigilance team, medical, and monitoring. And indeed, we focus on data quality and data timeliness. Here I display you our data flow. It's a little bit of busy slide, but if we start here on the left side of the slide, it starts when site makes a data entry for safety into electronic data capture system. So for EDC, we have access for CDMs, clinical trial managers, PV managers, PVMs, and medical representatives. We all do reviews for the data, and CRAs also can have a view to certain data. The red arrows reflect the interactions between our different team members handling and reviewing the data. So from EDC, serious reports are processed to our PV database, which of course then supports us in our submissions of SUSARs and ASRs to competent authorities. I give you here a quite basic picture of our serious adverse event data quality activities. Database, of course, it's tested and validated before we start and activate the trial. For SAEs, what we do at SA received is a triage. We check the minimum SA criteria if it's filled, data completeness, and also the timeliness, meaning if the SAE was reported by the investigator to us as sponsor in a timely manner. And if we find deficiencies or discrepancies, an SAE query will be addressed. For data entry reviews, we check the consistency of the data and the technical accuracy. We review every SUSAR before we submit. We check submission criteria, the content, and the technical elements, for example, E2PR3, so that it's valid for submission, for example, to EVCTM. For ASRs, we do a quality check focusing the content and format of the document. We have various reconciliation activities to review data consistency and also to manage if discrepancies are detected. For our TMF, we have a structural and scheduled TMF quality check on PV documents. It includes SUSARs and ASRs and also other PV documents like safety reporting plans, reference safety information, note to files, and other safety-specific documents. For data timeliness, we review essay receipt timeliness. We also review essay query response timeliness and submission compliance of SUSARs and DSURs. Here, I divided our activities like in two categories. Something we do on spot as an ongoing basis when we receive or handle the data. And periodic data quality activities are more like retrospective reviews. So indeed, as ongoing data quality activities, we have the triage at essay receipt plus timeliness check. We do quality reviews before submitting SUSARs and ASRs, and also at that point, we check that we are compliant with the required timelines. And for database, indeed, we do our testing and validation activities. For periodic reviews, we have reviews for AE and SA data. We have a periodic review for fatal serious related adverse reactions. There are reconciliation activities. We check the timeliness of essay query and SAE receipt. Plus, we review submission compliance for SUSARs and DSURs. And then there is this TMF QC for safety data and source data verification at monitoring. Our approach for safety data quality activities, this is a non-exhaustive list, but I wanted to highlight some things. We consider whether a full quality check for the data is needed or whether we could use a risk-based approach. We do self-checks by the data processors and, of course, also independent reviews by a second independent person. Our focus is in trials. For example, we create our DSURs, ASRs. Currently, we produce those trial-based, not compound-based. Our principles for risk-based approach is that if we use it, it should be justifiable, transparent, and documented. Our focus is in related serious data, meaning SUSARs and SARs, and also in fatal and life-threatening case reports and DSURs. Always the priority is in high-risk trials and in new medicinal products. We manage the possible findings and actions in our quality management system. I took here also a bullet point for internal process for data quality activities, because you need to have a process in place to do your data quality activities, and you need to maintain and keep that up to date. And adequate internal guidelines are indeed important. From technological side, we are moving more and more towards automated checks whenever it is possible. But of course, the findings are reviewed and actions managed by humans, by our experts. We utilize metrics produced by different systems like electronic data capture, pharmacovigilance database, and quality management system. Here in the end, I took a liberty to provide a few points to consider. Again, this is a non-exhaustive list, but I think it's important to keep in mind that non-commercial sponsors are a diverse set of stakeholders. There is very different types of organizations performing academic trials. The data volume is, of course, a very important driver, and it's good to evaluate the impact of your data collection approach or trial approach. However, in data quality and timeliness criteria, we usually cannot make compromises, and adjusted data collection, it indeed may reduce the volume of data quality activities. However, the impact might be minor or none when you are handling the quality of the SUSARs and DSURs. For data quality activities itself, we always try to think what is adequate, what is efficient, what is required, and what is doable. The situation of guidance for data quality activities is that currently, yes, multiple documents are available, as we already have heard. But when it comes to details and on how to do data quality activities, it may take some effort to translate the guidance or requirements to operational activities. What we also noticed is that in terms of getting trained to data quality activities, it might be not so easy to find suitable external trainings for non-commercial sponsors. That was my last slide. Thank you very much for your attention.
Thanks a lot, Tula. And well, questions? You can raise your hand if you have questions. Let me start for the question session actually on a follow-up on the last session. There was an impression that EMA is relabeling or reassessing the labeling of SUSAs. This is not the case. It's also not the case for the member states in clinical trials. We are assessing as member states for clinical trials, we screen and assess some SUSAs, and we come across here on some over-reporting, put it this way, where we could not find that it was unexpected or where it is rather disease-related and not compound-related, just to clarify here. So we are not relabeling. The safety reporting is really the responsibilities of the sponsors. But are there any questions?
Elke, I think that there was one as well in the chat.
I'm just scrolling. There's one on AI.
From Yvonne?
That's one?
Yes.
Yeah, from Yvonne. It's a question how to what extent AI is used by national competent authorities, human or veterinary side. Are there any feedbacks?
I can answer from the human side, from Austria only, more or less. So we are not that far yet, and there are discussions going on about how it can be used, but nothing definite now to in plan to be implemented soon, let's say. But I have been listening to the MHRA symposium 2 days ago, where MHRA actually presented their data, and they did implement already artificial intelligence and case processing to some extent, but I cannot really comment on that.
Thank you. At the veterinarian side?
Hi. Shall I try?
Sure, go ahead.
Thanks. So that was regarding AI, am I right? Yeah, so nothing in place yet. Multiple areas are being investigated for where it could be used, and it's definitely foreseen in the future to implement AI. For example, an idea that could be, and I think that is also investigated on the industry side, is having AI for case narrative reading and filtering to get more information that is mentioned in the case narratives, but not yet mentioned in data structured fields, for example, to allow for analysis on that end. So AI is in the progress, but we're not there yet. Thank you.
Thank you. And actually, same holds true for clinical trials. We are aware, we thinking about, but nothing in place yet. Tom, you have your hand up.
Yes, thanks, Elke. Two things. First, the second question from Enda, the second part of the question, she said if anyone was reperforming labelling assessments, would it be the expectations that we'd wish to receive all serious reactions? Absolutely not. No, not in SUSARs. Even if people are looking at the seriousness as part of labelledness as part of reviewing a SUSAR, please only continue to submit SUSARs. Regarding the AI use, the agency EMA isn't using AI per se, but we're investigating various ways in which we can do it. We've got a few little pilot projects ongoing. Our medical literature monitoring service, the MLM service, we're using some AI tools. Presently, we're only using them to target our QC. So we're continuing to use, let's say, natural intelligence with humans doing the assessments, but to try and focus our QC on some of the decisions so that we know where to look, because the team will review thousands of articles every day, and you can only QC a fraction of those. So to target that, we're starting to use it, and we're looking at a few other potential areas where we can do it. We're doing some natural language programming, which not AI in the sense of getting feedback and automatically changing the algorithms, but it's what many people would consider to be AI. We're doing some of that to tweak our duplicate detection algorithms. So once algorithms based on the structured data propose possible duplicates, then the NLP part will look at the narratives, because that's a tiny subset of all the potential cases, or potential couples, I should say, and see if it can adjust the scores. So there's a few little areas where the agency's doing it, but it's very important to note we're doing so very cautiously, and each step along the way is only taken after very strong evidence that the artificial intelligence is at least as good as, if not better than, natural intelligence. So we do tests looking for non-inferiority or superiority, and if it's non-inferiority, it's got to at least be superior in terms of speed. So that's how we view it. It's something which could produce very vast amounts of bad results, and so it needs to be very carefully assessed and made sure that it's well trained and at least as good as the humans doing it. Thank you.
Thanks, Tom, and thanks also for clarifying that we expect really only SUSARs to be reported and not everything. Sorry, we would not cope with it. I have a question here from Tony. They would like to know, can NCAs comment on the impact of compliance with GDPR requirements on the duplicate check with effectiveness? That's maybe also something for EMA or in the vets. I don't know who likes to. Katrin, first, please.
Thank you, Elke. Yeah, I can try. At least from our point of view, from NCA side, it is getting more difficult now, right? When we only have initials or not even a postal code, and that it makes things more difficult, especially for automatic detection. When it comes down to the manual progress, well, you end up calling the other sender and then trying to figure it out together by going through the report step by step. But for automatic detection, I would say we have experienced it becoming more difficult. Thank you.
And Anna?
Thank you, Elke. Anne Tobin here from the HPRA in Dublin. So I had a similar question around GDPR, but before I ask my question, I'm happy to give a comment from our side. The implementation of the GDPR legislation, and I suppose the requirement to protect the identification of the individual, has complicated the duplicate check piece. But I suppose the comment I wanted to make is it's not only complicated the duplicate check piece, it has, I suppose, impacted on many aspects of the ADR reporting system, in that it has resulted, I suppose, in difficulties on occasions on follow-up, difficulties in obtaining complete information on cases. Now, I do think that the implementation of the GDPR legislation is critical, because applying or ensuring anonymity of the patients, it does, I suppose, not impede on reporting. It supports the ongoing receipt of cases going forward. So that's just from our perspective. But I was going to ask a question on whether MAHs or CROs, etc., had experienced complications in their processes as a result of the implementation of the GDPR legislation. So I would be very interested in that aspect as well.
Okay, thanks. Would one of the MAs like to feedback? Maybe Jim?
Are we still on the AI question, or?
No, we are on the GDPR question actually, and the impact on our work of ADR processing.
It's we consider it a value add. I mean, it's that's all I can say about it right now.
So I think it has an impact to all. It's a valuable regulation, but we need to live it. And
Yes.
you had a feedback on for the EA. I think it's pretty in use at pharmaceutical companies, or?
We're using it. We're just getting, trying to figure out exactly its utility and where we can use it. But like was said by one of the other comments, it's used to compile things and put it in front of a reviewer to make the assessment. So we don't just take it, as gospel. But it is emerging as a pretty useful tool in many areas. And so we're tracking that progress, actively to see. There's certainly going to be applications in different areas of product development for sure. But it needs to be thoughtfully, like this is being addressed at national levels. It is something that needs to be, embraced very carefully.
Yep. Thanks. Tom?
Thanks very much, Elke. So, 2 points. As the guardian of a duplicate at VV, I've got a few comments on compliance with GDPR. A lot of what I see as people being compliant is, or trying to be compliant with GDPR, tends to result in over compliance, shall we say, where possibly a lawyer covering their back has said, oh no, you've just got to hide everything, and way too much is redacted, way more than can genuinely be considered to be personally identifiable information. A good example, a state or a town in America for a reporter, or in any city, the state or county, that level of information really isn't granular, really isn't personal, and can be provided and can make a significant difference in terms of being able to reject or confirm duplicates. I mean, again, to use America as an example, if you've got two cases which appear to be duplicates, but one reporter's from Florida and the other's from Alaska, you can pretty safely reject them. Another issue which I've noted is organizations redacting the data from a structured field. So they've clearly entered it into their database, and then with their overly restrictive settings, the data is redacted from the structured fields, yet it's still provided in the case narrative. So they're not even being compliant with GDPR because they're still informing us and making it available, but they're just not doing so in a useful way, which is one of the reasons why we implemented the NLP part of our algorithm for duplicate detection to try to counter that. So yes, that's an issue which I'd say has a big impact on duplicate effectiveness. So I would urge anyone to challenge what your lawyers say and to really consider what is actually identifiable information, and also to bear in mind the EV access policy, wherein we don't give out initials and date of birth, even in L2B cases. So this information can be provided to vigilance safely without the risk of it going further. Secondly, someone's actually, Nicola Gilson, hello, has put a question directly to me, saying that the top 10 to 20 senders are selected each month for individual case review. No, that 10 to 20 senders are selected each month. And what's our criteria? I'll be talking about that actually in session 4, and we use a combination of everything which you'll see in my slides in session 4 as our criteria for assessment. Thank you.
Thanks, Tom, and also thanks for answering what I would have asked next. Anya, you have your hand up too.
Yes, thank you, Elke. It's more like an observation. I think we have learned a lot about how all stakeholders are dealing with data quality and how much effort everybody puts into it. And I would just like to remind everybody that we also do a lot of really good stuff with all the data that we get. In all these discussions, we almost tend to lose how useful the data still is, and that we are picking up signals and safety issues. So just a bit on a positive note, after all the issues we've heard about data quality. Thank you.
Thanks, and thanks for putting it on the spot why we are doing it, why we're collecting data to find this as a safety profile, to see signals, to prevent adverse reactions. That's, I think, is all our job together and jointly. Yes. Any further questions, actually? Anne.
Thank you, Elke. Just one additional topic that hasn't really been touched on, and it's a topic that we do see in the HPRA. It's around the submission of reports for medical literature monitoring cases, where there's duplication of effort on behalf by the MAH for actives that are covered by the EMA system. So again, that results in duplicates. It also results in work on behalf of NCAs, and I know the EMA. So I do think it's an important topic not to forget when we're considering data quality. And before I give the floor to others, just to echo what Anya has said, yeah, there's a huge amount of good work being done. So yeah, not losing sight of that. I suppose it's always striving for continuous improvement is why we're all here. Thank you.
Thank you, Anna. And thanks for also pointing out the medical monitoring cases. Any further thoughts on the topic, or are you ready for a break now? So if there are no further questions, and thanks to all, thanks for the discussion. Oh, there's a question for Anja just coming in. Sorry. Opinion on reporting on individual case safety reports associated with product quality complaints associated with combination products to EMA. Is you meaning reporting if there's a combination and the ADR maybe to quality? Anja, could you take it? Just.
Thank you. I'm trying to digest the question.
Yeah, me too. So sorry. Thinking and talking is not always the best. Sorry to put you on the spot, but you were mentioned, and we often have the problem in combination products. Is it causality by the one or the other? That's what I know from clinical trials. But this may be here. I don't know.
I think for product quality, there are two routes to inform the product quality route, and you have the ICSR, let's say safety pharmacovigilance route. So I'm not sure what the.
So I'm also not so sure about about the content.
Yeah, perhaps if it's about let's say characterizing the data sets that we have in Eudravigilance, is this is the intention of this question perhaps that we should acknowledge that there can be product quality complaints or combination products in the data set as well? I'm sorry, I'm not sure I understand the question.
I'm also not.
Apologies.
Maybe Swapnil, could you update us? Maybe you were online.
And now we have given the right to talk. I think he can unmute himself or herself.
Yeah, no, sorry. Just want to understand the combination products, like a device and a medicinal product, a combination. If there is no adverse event associated with that, but something happening with the device, just like we can say like auto injector. Currently, we have a product like EpiPen auto injector. If any adverse event associated with the auto injector, but not with the epinephrine as a medicinal product, in that situation, do we consider to submit such ICSR to EMA? That's what my question is.
So if you have an ADR of the combination with a medical device, this needs to be reported to EMA.
Yeah.
Yeah.
Okay. I will come up with the. Yeah, sorry.
I don't know, Tom.
Yeah,
I don't know
think
who's.
device issues with no associated adverse drug reaction would not presently be reportable to EMA. We're looking at EMA is going to become responsible for medical devices, I believe, in the near future. So this will become an issue for us. But presently, in the situation that you describe, then it would not be reportable.
Yeah,
but
thank
if you
you
want to get
I
that
got
in
the answer.
writing, please do. As specific questions like this, the Ask EMA feature on our website, you can write to that, because this you just heard verbally. Now, if you need anything in writing, please do write to us. Thank you.
Yeah, thanks. Good. And you also need to qualify if the RDR is on the medical or on the medicines. So that also something, and it's a good note to follow up on with the EMA, asks EMA side. Okay, I think with this, thanks a lot, and thanks a lot for all your questions and discussions, and handing over back to you, Paulo.
Thank you, Elke.
Dear participants, welcome back. I hope you had a good lunch and a strong coffee to carry us through the last part of this wonderful workshop. And with that, James, over to you to introduce and get started with the next session.
No, thank you very much. And I also welcome you back from the afternoon session for this multi-stakeholder workshop. And I hope that everyone has had a nice break. So we move on to session 4 now, which is primarily a panel discussion, as you saw from the agenda on the screen there. And it'll be in the form of a brief presentation, followed by a substantial panel discussion where we've invited 6 panelists to contribute to that discussion. And then we'll open up the floor for comments and questions. And you can supply those in the WebEx chat function as the panel discussion is ongoing, so then we can lift those in if necessary. And in brief, the focus of the session is to discuss on how to tackle the challenges related to both recording, measuring, characterizing, analyzing, and improving ARD data quality. So it's quite a broad subject matter. So without further ado, I'll hand over to the main presenter of this session, which we've already met this morning, so he doesn't require much more introduction, and that is Tom Paternoster-Howie, and he also hopefully set the scene for the panel discussion and then for the questions later on. So over to you, Tom, and your 10 minutes starts now. Thank you.
Thanks very much, James. So yes, as James said, I'm just going to give a little presentation to hopefully stimulate discussion amongst panelists and get other participants in the workshop at your thinking. So I'll discuss EMA's position in the ADR data pipeline, challenges associated with being near the end of a pipeline, challenges related to recording, measuring, characterizing and analyzing ADR data quality, and challenges related to improving ADR data quality. Importantly, these slides, I'm not attempting to provide solutions to the challenges. I'm just trying to describe them a little and to stimulate discussion. And as Anya alluded to in session 2, EMA is not contacted directly by patients, healthcare professionals, or other primary sources. We're, to a large extent, at the end of the pipeline. All the data in EV comes to us via MAH's, NCA's, sponsors, or CROs working on the MAH's sponsors' behalf. And over the next 4 slides, I'm going to describe the routes by which different organisation types obtain and submit the ADR data to EV, and where you can see the kind of Chinese whispers type effect which can happen, but also issues where data quality matters can arise. So first of all, describing the arrival of an EEA ICSR via an MAH. So primary source, and I'm including literature even though it's kind of a semi-primary source, of course. Contacts, or is identified by an MAH or sponsor. They then send it to EudraVigilance. So MAH sponsor, we have a secondary source, we're a receiver and a tertiary source, and then we'd reroute it to the national competent authorities. So if it's come from an MAH in the EEA, a copy gets immediately forwarded to the relevant competent authority if they've chosen to receive it rather than viewing the cases in EV. And also they're available for MAHs via the L2A download, which a lot of people online will be very familiar with. So the next one, EEA ICSRs via NCAs. Again, primary source to the secondary source at NCA, or maybe via a regional pharmacovig centre. They'll send it to EV, and then it's not sent out to any other competent authorities. It's just made available to MAHs via the L2A downloads. So here we're fairly close. In both of these examples, we're just one step removed from the primary source. There's a primary source, someone's done coding, the data gets sent to EV. Although the recipients, certainly from L2A downloads, via the access policy, you wouldn't get all the data as we previously discussed. But now I'll look at the non-EEA ICSRs, which can have some interesting ways. So from an originating MAH, it's fairly similar to EEA. You've got your primary sources, you've got the secondary source, the MAH, and then the case comes to EV. Importantly, non-EEA not made available outside of the agency, not forwarded on to any NCAs, and not available via the L2A downloads. But I think here is an area which lots of people online will recognise as being a major source of potential issues regarding data quality is the non-EEA ICSRs via secondary MAHs. So primary source will contact an MAH, a secondary source, or potentially might just have informed a regulatory authority straight away. If they didn't, if it went to the MAH, when it goes to the regulatory authority. And then the MAH will receive the data via the regulatory authority. So already, the people who are about to send it to us are already a quaternary source. They're a long way away from the source data. At this point, of course, often many MAHs will receive the case at the same time. For example, if there's 10 MAHs who are both paracetamol MAHs in the USA and Europe, then all 10, when they get the case from the USA, would have to send it to EMA. And it's way more than 10 for paracetamol. So we end up with these massive multiplications at this stage. And then it's sent to Eudravigilance. But that's not necessarily directly how we get it, because either the MAH acting as a secondary source or a quaternary source can send it to their business partner, who then send it on to Eudravigilance as well. So the 10 MAHs could become 20 or 30 because of different licensing agreements around the world. And we can be at 5th, 6th step away from primary source. So when we receive an ICSR, there's various unknowns. What was the source information? Is all the reported information in the case? And would the sender have been permitted to include all the reported information, compliance with local privacy legislation? Was it accurately entered? Was it correctly structured? Basic fields must be structured to pass the business rules, but that doesn't mean that the structuring correctly reflects the information in the case or in the source data, nor even that it makes a valid case. And how many copies of the same ADR are in different cases in the database? Now, on the next slide, I've got a highly technical diagram which summarizes many of these challenges for EMA. You see here, it's highly technical. It's for Gizmotron 3000. And something comes in at one end. You see the top left. And then out the end, something else emerges. And if you want to do quality control, you're going to ask yourself a few questions. Who knows what it's supposed to be? What went into it? It passed the business rules. It's come out of the pipe at the end. So, yeah, we've got a little more structure than random shapes, but there can be certain interesting issues. Now, challenges related to recording, measuring, characterizing, and analyzing it. Scale of pharmacovigilance in Europe alone and globally can make it difficult to comprehensively assess data quality. We get around 10,000 ICSRs per day in Eudravigilance, and when the COVID vaccinations were in full swing, that went up to over 20,000 ICSRs a day. We get them from about 5,000 different unique senders of ICSRs per year, and some organizations will only send 1 or 2 cases a year and might not send for another year or 2. How best to prioritize assessments? And this goes back to that question which was asked earlier. These are some of the ways we consider. Random selection, number of cases per year, risk-based, looking at particular drugs of interest or having heard about new databases or whatnot, or targeted to coincide with other measures, or indeed use of the database level queries I mentioned, where you can assess levels of particular types of errors. Now, so those are some ways which it could be done, but there may well be many other ways, and I'm sure the panel will want to discuss about these sorts of matters. Now, other challenges, well, there's lots of different ways of characterizing quality of ADR data, which Anya alluded to in her opening presentation. There's completeness. Is everything entered that should be? Is more data always better? How many tests is too many? I bet there's quite a lot of people online who've seen some Japanese cases where you have 500 tests. And that might be slight overkill, particularly if the tests are many tests predating the ADR. Accuracy. Does the case reflect what was reported? What if the verbatim reported term was unhelpful, bordering on useless? A good example of this, again, going back to COVID vaccinations, in the first wave of COVID vaccinations, we started to receive a number of cases from a sender with the suspect drug of Pfizer. It wasn't actually Pfizer who was sending them. Thing is, the sender was being perfectly accurate. They were told Pfizer. They'd given out these particular kind of yellow card type forms to patients who were receiving the COVID vaccine. You know, in a COVID vaccine context, if you're someone who's just had the vaccine, Pfizer means only one thing, and everyone knows what that means. But in a database like Vigilance, Pfizer have, goodness only knows, probably at least 1,000 drugs, maybe more. And so you can't code Pfizer against Pfizer-BioNTech Tozanamaran vaccine, because then everything in the future would be coded against that. So we got incredibly accurate data, but incredibly unhelpful data in that instance. Also, we can consider structuring and unstructuring. Was the data being broken down into each structured field? Or conversely, was a beautiful long essay crafted lovingly to tell the story of the ADR and the narrative really well, but with a bare minimum of structured data, so you can't easily break down the case? Conversely, is the data all structured, but the narrative's just auto-generated with loads of redundant filler information like patient received drug for unknown indication on unknown date, and so on. And it takes assessors hours to read what should take them seconds. There's all these different issues, and how does one characterize these? And I'm glad I'm not having to write the Data Quality Framework ADR chapter linking that to overarching data quality framework concepts. Now, we've got one, I'm on my last slide now before I throw back to James. Challenges related to improving the data quality, because we don't have the ability to change any non-master or non-MLM cases in EV. The sender is the owner of the case and is the only one who can nullify it or change it. So we must request the senders to make changes, and that can lead to concerns such as conflicting demands. A case might be valid in one jurisdiction but not another, and your database might not be able to submit a nullification message without properly truly nullifying the case in your database. Do you want to nullify it and unnullify it? How do you explain that in the audit trail? That can be quite interesting challenge for certain senders. Company policies, internal SOPs and WINs may clash with EMA requests or requirements. You may need to take time to adjust those as well. And also, there's a very real, I've noticed with an MAH, and very understandable fear of messing with data from regulators. You received a data from a regulator, you've loaded it into your database. We've said, as a quality problem with this, again, you've got the nullification issue, but are you going to change the data? Maybe you know that it's definitely wrongly coded because you read the narrative, but are you going to change the coding without telling the regulator? Many MAHs really want to align their databases closely to the data that NCAs hold. So there's a whole lot of interesting challenges there, which again, I guess my colleagues on the panel will be delighted to talk about. So that's it. I hope I provided some food for thought. Oh, sorry, one other. I forgot my last bullet. Of course, the MAH may have sold the product and no longer has those cases in their database. It's someone else's problem, but they can't fix a case because they're not the sender of the original version. So we end up with an interesting problem there. So, as I say, I hope I provided some food for thought for everyone. I'll stop sharing now and hand back to James to take it with the panel. Thank you.
Thank you, Tom, for setting the scene. And indeed, there's a lot of food there for a lot of thought. So we will crack on. And what I tend to do is to invite the panelists to maybe briefly introduce themselves and provide their affiliation. And I know that one of the panelists we already know, but it's good to repeat sometimes. Their main affiliation, their sort of background. And if we do that systematically through the panelists, and then we can open up for the discussion, and I can pose a question at the end to everyone to engage in. But if we go in the order of what is on the agenda, I propose that we have Paolo Puscelli first, if he can if you can introduce yourself and provide affiliation and a little background, that'd be great. Over to you first. Thank you.
Good evening, everyone. I am Paolo Porcelli from the Italian Medicine Agency, and I am a GPP inspector for more than 18 years, more or less. I have experience also as a BMA assessor, GCP and GMP AP inspector in training. And so this is my position actually in the Italian agency.
Thank you very much. And then we'll move on to Rodrigo Postigo, please.
Hi, good afternoon, everybody. My name is Rodrigo Postigo, and I work at the European Medicines Agency in the Pharmacovigilance Office. So I represent here the group of people that are doing signal detection and validation using the Eudravigilance database and also supporting the PRAC on the signal management procedure. I've been working at the EMA in pharmacovigilance for the last 14 years, so I've been involved in many different issues, and previously I have also pharmacovigilance experience within the industry and also in clinical trials. So very happy to be here today. Thank you.
Thank you, Rodrigo. And then we move on to David Lewis, please. Thank you.
Hello, everyone. Dave Lewis. I'm the venerable age of 37 years in patient safety, so I think you could call me a veteran, maybe a dinosaur. I work for Novartis as QPPV. I also have a university affiliation as a visiting professor at University of Hertfordshire, specialising in pharmacovigilance.
Thank you, Dave. And then Magali Quentin, which we've already met, but over to you again, just briefly. Thank you.
Thank you, James. Yes, Magali Quentin. I'm working for Boehringer Ingelheim Vet Medica. I'm the QPPV there. I have more than 15 years of pharmacovigilance experience in industry for both veterinary and human medicinal products. Thank you.
Thank you. And then we have Joop van Geven, if that's correctly pronounced. I apologize.
Thank you. Yeah, no, that's fine. My name is Joop. Yes, Joop van Gerven. Thanks for the invitation, by the way, for this very interesting workshop. I'm a professor of neurology and clinical pharmacology in Leiden for the past 25 years. I've been a researcher, so basically a professional generator of what later came to be known as SUSARs for over 40 years now, I guess. And since about 11 years, I'm the vice chair and now the chair of the Dutch Competent Trial Authority, CCMO, which serves both as a trial regulatory agency and as a central ethics committee. So I have great interest in this workshop because it shows me, let's say, what the gizmotron looks like. I'm both at the end where everything goes in and the end where everything comes out, and I have no idea what the relationship is quite often actually between the two. So very looking forward to this panel discussion.
Thank you. And then finally, that is Bauke Rijmakers, if that's correct.
Thank you. Yes, my name is Bauke Rijmakers. I'm head of signal management at Uppsala Monitoring Center. But first, I was a medical doctor and I have about 10 years of experience in pharmacovigilance. First as an assessor at the Netherlands Pharmacovigilance Center, LAREP. And about 2 years ago, I joined UMC as a pharmacovigilance scientist. For those who don't know UMC, we are home to Vigibase, which is the WHO global database of adverse event reports for medicines and vaccines. So EMA was just referred to as the end of the pipeline or the end user. So that would make us the end user because in Vigibase also the European reports are collected. Thank you.
Thank you and welcome. Now, thank you for all your introductions and thank you for accepting the invitation to be panelists here. And it's obviously we've got a very broad background, so that should be good for discussion. So I suggest that we will start with one question, and I invite you all to respond to that one question so we can kick off the discussions, and that is, what does your organisations consider to be the most important or significant challenge regarding adverse drug reaction data quality? So quite a broad question, but what do you consider is most important and significant, just to kick us off? And I'm not sure, we don't have to do it in the same order, but please just raise your hand if you want to start off. Unless I force you to, potentially, Paolo, you could start because you introduced yourself first. Maybe you can start.
Yeah. I think that for our side, the change it's not big, because from the inspector side, we have just to check and review what the MAH put in place in order to follow the guideline and indication coming from the EMA. So I think that we have seen during this morning this example, what the inspector checked during the inspection, and also in the last presentation coming from Tom, we have seen where EMA stopped when received the ICSR and the various unknowns. So the inspector have the possibility to capture some information in relation to the source, to the reporter, and so also for the training of the people that are involved in this kind of activity from the MAH. And so I think that we from our side, we follow all the new indication that we will arrive from the challenger.
Mm-hmm. Yeah, thank you. And from your inspector's point of view, what are the most common errors that you see in relation to adverse event reports? As in, what are the common issues that you see, or errors that that you come across? Are there any that stick out specifically?
From our side, we check, of course, we start from the training of the people, and we have a focus on the database in order to check the data quality. And sometimes we are in close relation with the EMA, with the agency, and we share information, especially during the inspection of the pharmacovigilance system, where there is at least a medicinal product registered with a centralized procedure. And in this case, as example, we can share with the EMA the review of the ICSR data submitted to EU Vigilance. In these cases, the inspector verifies some cases and evaluate the related activities and the tools available of the MAH. And the review consists of three important aspects: the quality data review of the content of the ICSR, the recording of the medicinal product information provided in the ICSR, and the potential duplicated cases identified. Plus all the things that we have discussed in this morning. For example, if they to align the database to the close national competent authority, that is a big issue when you comes to the MAH. And so we check the possibility that they have to change something in their database, even if they cannot ask a change in the national competent authorities database.
Thank you for that insight. And maybe David, do you have any particular sort of issues that are of significance that you consider in terms of data quality?
If I try and quantify in simplify it into just a few words. It's the medical integrity of the report that we submit, which has got to remain faithful to the source data. So if I'm considering from a holistic perspective, it's got to be accurate, it's got to be complete. We should report in a timely manner, and what we report should have a clear structure. I could go into a lot more detail because every report, as we've heard, has its own challenges. From the clinical trial cases, which are generally well documented and from experts, to some of the reports that we receive from patient support programs, obviously initiated and written by a patient. And there are challenges inherent in that. I'll pause there and let others speak, but for me, it's medical case integrity.
Yeah, thank you. No. Anyone else, any panelists want to come in spontaneously? Or,
Yes, if I may.
Yes.
Well, my major concern as a chair of an ethics committee and now also, but to a more limited extent as the chair of the competent authority, is the quality of SUSA reporting, basically. Now, of course, we have many very insightful adverse drug reports and annual updates and other types of overviews, which are terribly insightful. And but if I get SUSA reports, which I still regularly do when we are safety assessing member state, for instance, I'm always disappointed by the quality. If any of my clinical trainees, and I teach and educate neurologists and clinical pharmacologists, if any of them would dare to send in a clinical letter to any of the in our healthcare system to any of the other physicians, I would fire them immediately. I mean, these reports are completely, they lack any comprehensible structure. They lack any insight into what is actually going on. They seem to be a collection of everything that is probably relevant, but because many times people are afraid to decide what is relevant or not, put in just about everything. So that for me really sometimes gave me sleepless nights, simply because I didn't know what I was supposed to do, because I didn't know what was going on. Apart from the administrative burden of teasing out all the separating one report from another and finding whether it's an update or something new, or which has all been covered and discussed in this meeting also. So I think knowing that these SUSA reports are really quite terrible, I can only applaud the Gizmotron technicians who actually managed to turn that into something useful. But I'm really concerned about this input, and it has been mentioned more often. But I think the main reason why this input is bad is because the physicians don't really know how they should report, and there is a sort of uncertainty and perhaps even fear from the position of sponsors to be incomplete or to be directive, or and I think this is a terribly dangerous mix of information. I have other concerns about the process as well, which I probably will bring in later, but I think this was one.
No, but thank you. In relation to what you just stated, as in, is there any sort of steps that could be taken? As you said, you alluded to the fact that it's probably an education is required for the physicians, and then the relationship between physicians and sponsors. Is there any way, have you thought through the process of how to improve the scenario potentially?
Well, for one thing, I've never really understood why the sponsor should and why the report of the physician and of the investigator and the sponsor's assessment should be put into one document. That confuses me terribly. So that may be one way forward. I also know, of course, that the physicians themselves they are forced to put their information in some format which is completely unnatural to them, unless they're of course highly educated and gradually become more experienced. So I remember my initial experiences with this, and this by itself made it difficult to actually write down what I thought was going on in a particular case. So.
Thank you for that insight. And then I'd like to invite the other panelists to come forward, as in either to address the initial question of what is the main significant challenge in your organisation. As in, to be fair, I don't maybe who have we not slipped in? As in maybe Magali from the veterinary side, or Balku, who's put a hand up. You can fight over it, but maybe Magali is, and maybe you can come in. Although you've already presented quite extensively the challenges you have already, but please reiterate. Thank you.
Thank you, James. Yeah, I think for us, and indeed I have presented it, I think that this harmonization internationally, so between Europe as a region and other region, is the challenge for us in the vet industry. I would go a bit further now in this discussion and say we see also divergent preferences inside Europe amongst agencies, which is also a challenge for us. With different requests towards the way the case are presented, the data are structured. So that would be my big word to summarize the challenge we have is the lack of harmonization.
Thank you. And have you got a suggestion of how to a step to combat that? As in, is it down to just the interpretation of the guidance that's existing, or does it mean that we have to really elaborate on the guidance so it's more strictly and less easy to interpret, so it's more strict? Thank you.
Well, what I see, at least for Europe, yes, I would see it's a divergent interpretation. It's a preferences. It is perhaps historical. It may be also driven by a particular product that a rapporteur may feel more close to, and then scrutinize the quality with certain eyes. And then putting the request differently. I think otherwise, from the level of guidance, at least in Europe, we should be fine, where we have also that this harmonization, and that is what was my message, is at the VICH level, so that we can have at least a common cases to propose to all agency that meets a minimum of quality. That's also a point.
No, thank you. And now I'll release in Balku. You'd like to come in? Thank you.
Yes, thank you. About the most important challenge with data quality, I would say for UMC, it is the missing information, and this can be either missing from the start, so it was not reported in the initial report and not obtained in follow-up efforts locally, or it was available locally, but it was lost along the journey to Vigibase, and this can be unintentional for technical reasons like coding or mapping problems, or it can be intentional, the masking of certain aspects to protect the privacy of the patient, for example. And most importantly, in that category would be the narratives. If they are not shared with us, that really loses the context for us to interpret the case. So I would say that's the most important challenge that we have. Thank you.
And because I'm aware of UMC, because I'm based in Uppsala as well, and the fact that you don't get the narratives is quite a significant issue, because you can't really judge whether the coded information is correct or not. And obviously now you've suggested there's missing data, so that's interesting to know. But and the reason why you don't get the narratives is
GDPR.
GDPR. Okay.
For example, but that's in Europe, that's the reason. Yeah.
Not that they're in Swedish.
And then Rodrigo, as in, I don't want to leave you behind, as Emma, as in, would you like to come in and give a perspective as well?
Yes, I have my hand raised. So yes, I wanted to give a bit the perspective of the EMA. This morning I was taking some notes about the aspects and the data quality issues that we face at the signal management group at the EMA. First, I want also to acknowledge the data, because something along the lines of this morning, it is about 12 years now of the PRAC, and I do not remember one single month without signals in the PRAC agenda, and it is over 75% of those signals are coming from spontaneous reporting systems, either from Vigilance or from the national competent authorities databases. So there is a lot of value in this data, and the data is really helping us to identify new risks with the drugs. Nevertheless, we are in a data quality framework, so I would like to highlight some of the issues that we daily face. Many things have been already said, but the duplicates are important, and they create a burden for us, because the amount of duplicates can immediately, when we do statistical signal detection, can reach the drug-event combinations can reach the threshold for a signal of disproportionate reporting. But then there are plenty of duplicates, and I think during the years we have put a lot of effort in the management of duplicates, and maybe it's the time to reconsider whether we need to put some efforts trying to avoid those duplicates from the very beginning. And I would highlight two different aspects. One is the literature, and part of it is trying to cover this part is the MLM service at the EMA, but there are still many substances that are not subject to the MLM, and therefore that can create an incredible amount of duplicates, especially when there are literature articles that they refer to other literature articles, and we have seen terrible things, cases from non-Europe enter as European cases, because at some point those cases appear in a paper that was published in Europe. And another source of important level of duplicates are the release or the publication of the data by the different regulatory authorities all over the world. We have increased the transparency, providing the marketing authorization holders with the relevant cases, but that has increased enormously the number of duplicates. And sometimes is part of the legislation, and duplicates are one of the characteristics of spontaneous reporting systems. So, but something to consider. I will not talk more about duplicates because many things have been said. The lack of information in the cases, especially for signal validation, is very disappointing, because we wouldn't raise a signal, or we wouldn't think about validating a signal until we have a case series that at least that is considered that the signal requires further analysis. And we do have a lot of cases with a lot of lack of information, and especially patient support program cases are not very useful for signal validation. They're useful in terms of numbers, because they help us to reach, the level of a signal, but then for signal validation is not very useful. I want to talk also about the follow-ups. And not many things have been said, maybe, but follow-ups are very important and necessary, because they provide us more insights into the diagnosis and more insights in the test. And sometimes with the first encounter of the healthcare professional or the patient or the principal investigator, you don't have enough information. But follow-ups are very, very challenging. And we know how difficult sometimes it is to get follow-up information. And I would really welcome more efforts in order to get as much information as possible within the first encounter with a healthcare professional, the principal investigator of the patient, because then we know how difficult is to get the follow-up. So, and also very important, it has been said already many times, in human and vets, the narratives, and a bad narrative can invalidate the case. And the narratives have a lot of impact regarding the follow-up. And I mean, the good thing about the EMA and Vigilance is that we read cases from many different marketing authorization holders, so we see different styles and different strategies when the data is entering the narratives. And something that really, really worries me is the companies that do not integrate properly the follow-up in the narrative, and they write one narrative after another depending on the follow-ups, and you can end up with 6-10 different narratives, and it's totally useless for an assessor to start reading information that is going to be corrected further down the narrative. And on the opposite, we see good practices as well, and we see companies that they do not copy one narrative after another, but they integrate the follow-up information in the narrative, and therefore is more clear. Regarding narratives as well, because we do still rely a lot in the narratives for signal validation and then for signal assessment, is crucial for us that the narrative is coherent with the electronic information or with the information in the electronic fields, because when the information is not coherent, we don't know what to believe, and sometimes that invalidates the case. So a proper description of the narrative of what we see in the electronic forms is also crucial for us. And there are sometimes we need to also take care of special considerations, like we're talking about interactions or medication errors, special situations like off-label misuse. Sometimes these things are coded in the ICSR, but when you read the narrative, you can't find the medication error. You don't know what interaction they're talking about, or you don't know what kind of off-label you are making. So it is very challenging. And I just want to talk about something, and this is my last point. I apologize, and it's the sender's comments. And we do read the sender's comments, and the sender's comments should kind of reflect the assessment, in this case, of the marketing authorization holder. And sometimes at the EMA, before we think about raising a signal should be something new. So we look at the PSURs, we look at the RMPs, we look at variations, and we also look at the sender's comments. And sometimes one of the considerations we have for raising a signal or not is whether the reaction has been already discussed somehow by the marketing authorization holder. And within the sender's comments in the ICSR, that give us an idea whether the company has considered the possibilities for causal association. And it is sometimes disappointing to see those sender comments, like sometimes we feel it's even copy-paste from one case to another, and there is not much assessment on those senders' comments. And I have to say, pharmacovigilance now goes, apart from the causal association, we also need to identify specific circumstances or specific patients that can be more vulnerable to adverse reactions. And it's sometimes disappointing to see that because someone is an elderly patient, that everything could be confounded by the age, or if it's a patient with a cancer disease, everything is confounded by the cancer. So it's a little bit disappointing to see in these senders' comments, like the proposal from the company is, well, this is polymedicated patient, elderly patients, cancer patient, patient with diabetes, they can have almost everything. So sometimes we raise signals because we don't see a kind of thorough engagement. It doesn't mean that the engagement is not there, but it hasn't been fully demonstrated, with the sender's comments in line with what it says in the PSUR and if it has been discussed or not. And I'll stop here. Thank you.
Thank you, Rodrigo. And I'm David. You've been waiting a while. You come in here. Thank you.
I'd just like to address a couple of the points raised by Rodrigo, because it's a common empathetic feeling that I can express. I despise narratives that don't give me a clinical story. I need to have chronological order, organization in the narrative. So absolutely, Rodrigo, I despise narratives that are broken by follow-up, contain contradictory information in different places. So what I would say, the best clinical narrative is one written from a physician's perspective, from the attending physician. So it tells a story, it relates it consistently, and it presents it in a very obvious chronological order. I just ask for one piece of empathy from the EMA side. Bear with us, because from the industry side, follow-up is remarkably difficult. And I'll just illustrate with one example. So every literature case obviously has an author for correspondence. Very easy. We can follow up with that author. Well, I surveyed 1,000 literature articles. We sent a follow-up to all 1,000 authors for correspondence. We have had 3 replies. That's the level of feedback that we get. And clearly, we're not here to take time to bite the hand that feeds us ADRs, but we are trying to target the follow-up so that we ask questions rather than just say, give me everything you've got. But it takes two to tango. If you want a good case report with appropriate follow-up, target the questions, but we do need answers. And I've published on this, I'm happy to put in the chat a reference. That's not the commonality. Not all case reports receive a fraction of what we'd hoped in terms of follow-up. But our follow-up on a good day is somewhere in the range 30 to 40%. So the majority of cases are actually lost to follow-up.
Thank you for those numbers concerning. And then Magali, as you want to respond as well, please.
Yeah, and I would emphasize the same issue at the vet industry than Dave just described with the follow-up. We encounter the same issue, and somehow it's a bit of a relief to hear that it's not just due to the structure of the vet business. Means we have to try to motivate a vet who has just to run his clinic, to take the time to answer our questions. One way that is used in country like US, for example, is you propose a customer care program where for reporting detailed information on an adverse event, you will get some financial support from the companies with regard to reimbursement of a product or reimbursement of diagnostic for the animals. But this is very limited. This is not allowed in all countries because of local law. But that's a way for us in the vet industry to get more thorough follow-up indeed. But that's linked to this commercial aspect that I've spoke about earlier. Thank you.
Thank you, Magali. And Joop.
Yes, well, of course, we've all lamented about the quality of the information that we get, but there is an inherent structure that is quite unequal in the information and the data that are submitted to us. They actually range from highly detailed and sophisticated, in the best cases at least, analysis of information related to drug effects, like in early clinical trials, or where there's lots of mechanistic additional data that actually suggest a pathogenic or pharmacological or toxicological relationship. Background information about the class of drugs, additional information about other similar agents can then be taken along. That's, let's say, the most detailed, and I would argue also most valuable information about potential drug effects. Now, the problem with that is that of course you lack numbers to know precisely how significant this is, and there's always a case that it's a flaw. And then, of course, at the other end of the spectrum, you have information from phase 4 pharmacovigilance programs and information after market registration and clinical use from public sources or healthcare professionals, which are inevitably going to be of much poorer quality. Now, my question really is, how is that level of detail and these differences in information that you have accounted for in the analysis by your gizmotron? Is there some sort of Bayesian approach, or perhaps even more desirable, is there sort of a mechanistic approach to accumulate data based on a meaningful signal to start with? That's simply something I don't know, because I'm not part of that machinery.
Yeah. Are there any responses from the panelists? I'd also like to, because of time, like to slip in some questions from the audience as well, and participants. I'm not sure that there's a few that have come in, but I'm not sure if they're going to break up discussion, unfortunately, because they're probably not in sequence. But there is one question here from from participants saying that are there any regular interactions between authorities and stakeholders on data quality on the human side, apart from individual case basis, as Tom has described? As in, can anyone respond to that one at all?
Well, I can only speak for our own country, of course, and that is something that we have highly desired for years, but that turned out to be terribly difficult. I mean, part of my question is related to the fact that we have different organizations responsible for different parts of data collection, and we all have our own specific expertise. My expertise is in pathophysiology, pharmacology, toxicology, which is quite different from the epidemiological approaches taken by my colleagues at LAREP, where Bauke also used to work.
Thank you. And then Dave?
James, yeah, I can talk to a direct interaction between regulators across Europe and industry in the Web Radar Forum, which was an IMI-funded project. This was really focused on the value, or potential value, I should say, of social media case reports and trawling social media to see if we could find a valid source of adverse reaction reports. And there we did engage with patients, healthcare professionals in a general assembly discussion. After around 3 years of research, really our conclusions were, this is noise. If I liken it to listening to a conversation in a pub, that was pretty much what we were seeing. All the bad behaviors, all the good behaviors, but no coherent picture of even a single case. And yet, we are still legally obliged to collect social media reports and to file them as spontaneous, i.e. unsolicited records, when we pretty much have determined that there is minimal value. Going back to something Hope mentioned, which I very much agree with, I think there's huge value into looking into mechanisms, and mechanistic safety is definitely a way forward. You aren't going to get that from social media cases. But SUSARs potentially, yes, a very rich source. And I would add in, it's not just the phenotype, it's as we progress with pharmacogenetics and pharmacogenomics generally, there is a huge source of value and data. We're going to become more and more sensitized to collecting very personal pharmacogenetic data. Another thought.
Thank you, Dave. Any other responses from the panelists at all? And otherwise, we can take another question from the audience, so that they feel like they're participating as well. And here's a question from the floor as well. In terms of spontaneous reporting for human medicinal products, which message should be adopted, quantity or quality, considering that a report is valid only with 4 minimum criteria? Isn't this an open door for low quality reports? And I'll stop there for that question. So quantity versus quality, and that a valid report is only based on 4 criteria. Is that not no? Anyone wants to respond with that one? Dave, thank you.
Sorry, I'll go for it because I've been part of the ICH E2D R1 working group, so the post-marketing focus. And the answer is both, I'm afraid. We need a high quantity because we will detect through spontaneous reporting rare, potentially very rare signals. If we look at the vaccine-induced thrombotic thrombopenic syndrome with COVID vaccines, we could not have detected that without the massive vaccination campaigns. The practical upshot is, what over 14 billion doses of vaccine generate signals. And the poor quality of many of the reports is almost taken care of by the fact that the well-documented reports, even though they're few, are added to by the noise of the poorer quality. And together it does yield a stronger signal. So it's both quantitative and qualitative. And the case reports that we would describe as those of high medical importance and potentially high medical impact have to be the ones where we focus on the best quality. But give me volume 2, I can work with both. the statistical methods are really powerful that we adopt these days. So the drug event combination, all based around Bayes' theorem, works really well with high volume. And it drives us to focus on the best quality to determine the nature, the mechanism of the signal.
Thank you, David. And Joop, please come in.
Well, I have a question actually. How much use do we make of drugs that never make it to the market? Because they often fail because of adverse drug effects. And if you can relate this to a certain mechanism of action or anything specific to that class, that may actually be highly relevant for potential drug effects for compounds that do reach the marketing stage, which are far and few in between. So I wonder if there's any cross-fertilization between compounds, which of course sponsors may object to, and which should be taken very carefully. But one brief example: we have seen well over 130 different antisense oligonucleotides and other RNA therapies, and they're currently registered about 5. So the adverse events in with these compounds, I mean, we've seen them coming for years before actually the first one hit the market.
Well, that's an interesting concept, as in it's almost like a pre-authorization profile that you'd have.
Yes.
Yeah. And Bauke, as in you'd like to come in.
Yeah, I wanted to say that I agree with Dave, what he said, that you need both quantity and quality, because without quantity, it would not stand out statistically, the combination. And quality, you need to take it further than just detecting a signal of disproportionate reporting. So I would fully agree with what Dave just said.
Thank you very much. And then Paolo.
Yes, I agree with the colleagues, but I want to just add that I believe that we must take into consideration the type of medicine, because for new medicine, every type of information is important, and therefore both the best quality and the quantity are important. For medicines that have been on the market for years, it is important to focus mainly on quality, because we know a lot of things in relation to the drugs. In fact, during the period, the pandemic period, we have seen an increasement in cases, but with a low level of quality. And so we connect with the other discussion on the follow-up. We have seen a lot of follow-up that given the possibility to capture more information for signal and other activities.
Thank you, Paolo. And then we've got limited time left, but I'll just take another question from the chatters, and the next one on the list is: have the marketing authorization holders analysed whether there's been an impact on the quality of cases when registration and coding has been outsourced to countries other than the countries of origin of the ICSR? Has anyone got any experience of that, of outsourcing, and any impact on quality?
Yes. I apologize, James. We are there. Most of the big pharma companies are outsourcing vast numbers of cases. I'm not going to talk about specific continents or even countries where much of the outsourcing is done, but predominantly all those countries clearly are English speaking, because English is the harmonized language for ICH style reporting. What do we check? Well, certainly from Novartis, we will check all of our vendors. We pre-qualify by audit. We look at individual case types. So we're looking at clinical trials, non-interventional studies, registry cases, patient support. Everything is there. How do we check the quality of vendors? Well, my company may be different from others. We look for what I call major errors, which I would describe broadly as medically significant, and minor errors, anything that is not medically significant. So what is medically significant? An error in seriousness, an error in causality, an error in signs, symptoms, diagnoses, labs, medical history. What is non-serious, non-significant, I beg your pardon, a typographical error in a narrative. It's not going to change the meaning of the case. So how do we record the errors? Well, we sample. I use the ANSI tables, which are really based on quality sampling of finished product. But you can get a good measure from the GMP ANSI standards of how many cases you should sample to find routine systematic errors. And I'm then asking all the vendors to be checked in-house. We check how many errors per case. And that sounds appalling, but I'll say medically significant errors, we're getting 1 error per 20 cases, which I think is reasonable because we're trapping the errors. For the typographical type errors, it may be as high as 5 to 10%, usually in that range. We even track errors per tab card. What do I mean tab card? Our database has a tab for patient, tab for products, events, one for narrative, one for medical assessments. So we're driving quality at every possible level and recording with the intent of either providing incentives for good quality or penalties for vendors that deliver poor quality. So hence we try and manage the vendors to drive for better quality because they earn more. It's the carrot rather than the stick that I prefer. Hope that answered the question.
Thank you, Joe. I think it probably did. But unfortunately, it feels like we've only just got going, and it's always the case with these panel discussions. You need a little bit more time. So I apologise for abruptly calling a stop, but it is 3 o'clock and we need to move on to the next session, which I'm not chairing, so we'll need room for that. But I'm sure there'll be opportunity to carry on discussions in the next session because that is also a discussion. So, but I thank you all the panelists for their time and accepting the invitation. It's been a rewarding, and thank you for the questions in the chat so we can have some engagement with the participants that are online. So I close this session with 2 minutes over and hand over to whoever's chairing the session 5. So thank you for me for now. Thank you.
Thanks a lot, James. We are ready for the next session, and Anne Tobin will chair. Anne, over to you.
Thank you, Paolo. Thank you, everyone. So I'm happy to moderate session number 5. So this is, I suppose, the last of the formal sessions today. The wrap-up comes after me. So just to introduce myself again, I briefly introduced myself during the Q&A earlier on. I'm the pharmacovigilance manager here in the HPRA in Dublin. So in this context, my key responsibility and that of the team that I work with is around the management of the national ADR system here in Ireland. So encouraging reporting, reviewing the data, processing, follow-up, etc. And I suppose quality is an absolute key aspect and a key drive that we do here in the HPRA. I also work closely with my colleagues who work under the guise of the PRAC activities, so that we get constant feedback in terms of the added value that spontaneous reporting is to the activities of the PRAC and vigilance assessment, and also work closely with our safety comms team. So I suppose to delineate what I see the difference between this session and the last session, there's a lot of commonality in that. I hope the discussion will continue. So this session is really to give a voice to all participants, so speakers, panelists, and everyone who has joined today to listen, to give, I suppose, continue the discussion, to collate feedback. But another key aspect of this is supposed to be more focused on what we think would be the key elements that need to be captured in this chapter, in this ADR chapter under the data quality framework. So during this session, I hope that we'll be using tools like Slido, even though I can't quite see the link to it on my screen, but I'm hoping I'll be able to see it soon. So we'll be Q&A, some discussions, and capture the feedback on some specific questions. So as I said, happy to continue the discussion, but if we could really be focused on, there's the slide on now, really be focused on what we think are the key points that should come up in this chapter. So I assume people have to use the code or the QDR there to link in, and then use the passcode to join and be able to vote on questions. So hopefully we'll have a first question up soon. Yeah. So our first question is, do you think that Data Quality Framework ADR chapter should include a section on the impact of duplicates or duplication of cases? Oh, wow. So far, I'm not sure how many votes constitutes 100%, but 100% of voters so far have voted yes. So we'll just wait to see if any no's come in. Oh, a few no's. So duplicates and the discussion of duplicates has come up throughout the sessions all day. And I suppose to me, a lot of the issues raised, I suppose, would form two different categories. So the challenges around noise, so noise created by underreporting, over-reporting, reporting biases, and duplicates. So to me, a key element of the chapters going forward would be around consideration of duplicates and the impact that duplicates have on the quality and of the data. I also want to take an opportunity, if anyone wants to raise their hand to ask any questions as well at this point, or if any of the panelists or speakers or anyone of the participants wants to come in with any comments. Okay, thank you. I see now that we have 54 voters in, so I don't know how many participants we have, but oh, they're still coming in slowly but surely. We can leave that go on in the background. I am going to carry on from a question though from the last session. So, and I think it could feed into this topic about what should be captured in the chapter. So it came from Enda and it related to the UK MHRA previously worked on a guide to support consistent safety case processing, coding, and improve first-time quality of ICSR received. So would a similar document, which is more than the E2B or 3 implementation guide, to address some of the key points raised by Tom, be of interest to the EMA? So I suppose when I had read the data quality framework, I was finding it hard to, I suppose, delineate between what this ADR chapter would contain versus what the current guidance would contain. So I don't know if there's somebody from EMA or people from the data quality framework who might be able to, I suppose, give further clarity on that delineation.
Hi, Anne. Sorry, I'd raised my hand, but I don't think you'd been able to see it on the screen. So yes, this document which we're working on, it won't be quite purple guide or green guide, but we're hoping that it will help to feed into overall data quality practices. It's going to be more in the terms of the quality assessments and the work that's done on the quality rather than directly on more granular details on how to code cases, because we're not building such a document, but it would be interesting to consider the possibility of such a document, because I remember the MHRA guides and they were really quite useful back in the day. Thank you.
Thank you for that, Tom. I suppose I have a few screens going here, so please prompt me if I'm missing hands. I don't see any further chats or questions in the chat, or I don't see any other hands. Oh wait, I might see a hand raised. Dave, do you want to come in there with a question or a comment?
It's a comment, and I've got to be a bit careful. I apologize. I've reviewed what I view as probably the best paper I've read on duplicate checking, which I know is under review at the British Medical Journal. So just to prepare everyone, there is a superb publication I hope to be released later this year. And I stress I'm a reviewer. I cannot mention names or the source, but there is a very, very good paper about to be produced, which reviews really the last 30 years of challenges with duplicates. And their review is comprehensive. It's very concise. And it does make, I think, really excellent recommendations about improving duplicate management and eliminating some of the problems. If I just extend that briefly, there is also another very good publication by John van Stecklenburg et al. On a new phenomenon or a newly recognized phenomenon, which is described as case replication. And this is a phenomenon that I think Rodrigo referred to, and our WHO UMC representatives will recognize, where the same case is processed by 2 or 3 different sources, quite often license partner companies. Each applies their own in-house process. So the same patient and the same report looks different in every way because each company processes it differently. And I can put that reference in the chat, but it clearly illustrates, on average, one patient report is replicated 4 times. And in about 12% of cases, there are 10 replicas. So I think in the next year to 2 years, we'll be talking about replication removal as well as duplicate removal.
Thank you very much, Dave. Those statistics are, I suppose, concerning, but it's good to have, I suppose, a sense of the magnitude of the noise that is caused by duplicates. And I would say I very much look forward to that paper being published. I'm going to come to a question from the chat. So it is there an analysis showing what the main 3 sources of duplicates? How can this be targeted to solution, e.g. best practice guidelines similar to the MHRA? I don't know who would be interested or who would like to take this question. I don't know if any of our panelists have any feedback in relation to that.
I can take it. I don't mean to monopolize though, Anne, so if you'd rather go somewhere else, please do.
No, I'm happy for you to take it, and I suppose just to obviously very interested as well to hear that feedback. But there is a poll going on in the background as well, so Dave, take it away.
Okay, main causes of duplicates. One was already mentioned, and very deliberately, it's one that we all recognize: multiple license holders for the same active substance. So the example was perfect. If a literature case appears with a case series, let's say 3 patients, and if we take, I think it was Tom's example, rapacetamol, let's call it properly paracetamol, I think it's over 1,000 marketing authorization holders reporting 3 patients in one literature case. And if you multiply that every record in each ICSR will look slightly different, that's where we get both duplication and replication. If I cite the paper that I've read and reviewed, 87% of literature cases on FDA FAIR system are duplicates. If I give you the background rate of duplicates on the Novartis system, it's about 0.5%. So literature cases are a veritable nightmare. Other causes of duplication are the very obvious. For well over a decade now, we've sanctioned and endorsed, even encouraged patient reporting. So we're now seeing more patients report, as well as the pharmacist, the nurse, the physician, the healthcare professional. So we're actively encouraging duplicate reporting from both the healthcare professional side and from the patients. And I'll stop there, but there are many, many other causes of duplication.
Thank you very much, Dave, for that input. I would say, based on the poll and the discussions, the significant discussions about duplicates, to me it's quite obvious that the data quality framework needs to include, and I suppose, considerations around duplicate management. So at this stage, I think we'll take that as a given. Just in relation to the poll that's on actively at the moment. So I think participants were about halfway through voting, and it's around, do you think that the data quality framework ADR chapter should link the processes detailed in the ICH guidance and GVP modules to the overarching data quality framework? And we have a 94% yes. So I think participants in this workshop are very much in favour of that. So just moving in then on from the duplicate discussion, there's quite a long question from Enda in relation to GDPR. So I won't read out the question in detail, but I suppose GDPR as an impact on data quality. Obviously, we're all advocates of GDPR and the principles that it upholds. Is it a consideration for the data quality framework chapter? There was discussion before lunch on GDPR, but do you think the quality framework would benefit from chapters addressing or sections addressing this? I don't know, again, if any of the panelists or any of the participants or speakers would like to talk to this topic. Sorry, I checked my hand up here again. Sorry, I'm not sure. I can't see if there are any hands up. Okay, well, just from a National Competence Authority approach, while we welcome GDPR, we do see the challenges that it can pose in terms of, as Tom said earlier, increased redaction or redaction beyond that is necessary for GDPR purposes. And also as well, it does on some occasions impede the possibility to follow up. So from my perspective, I do think that it would be beneficial to have, I suppose, that as one of the considerations in the chapter. Sorry, there's a few different feeds coming into me now. Yeah. There's another question up on the Speedo or the Slido. Do you think the data quality framework ADR chapter should consider the different uses of ADR data separately? So this one, the poll is a little bit more divided. We're kind of nearly even Stevens between no and yes, no being regardless of the end use, high quality ADR is necessary in all circumstances. And yes, each use case has unique challenges, and I suppose approximately 10% are unsure. So for the drafters of the data quality framework, that question is a little bit more on the fence. I don't know how beneficial, or I suppose the feedback, all feedback is beneficial, but I'm not really sure how much that is going to help. The next question then is, my organization has robust processes in place to ensure good quality ADR data. So we have a significant proportion of the participants are either agreeing or strongly agreeing. Again, it's 60% agree and 30% strongly agree. I have a hand up. Tom, do you want to take the floor?
Thanks, Anne. Yes, I just wanted to answer the question about the GDPR communication, which you were just mentioning earlier. It's important to note that in that context, we were referring to data obtained from EudraVigilance being forwarded to third country competent authorities. And the context of it was that information CEDA in the US, which is not the FDA, but also under the Ministry of Health, or I forget what it's called, Department of Health, they were releasing quite a lot of our data. Cases were being sent, and then there was information which was more geographically available. Whereas our discussions were about what you can send to EMA safely, European data, in terms of complying with GDPR, sending European data to EudraVigilance. There's not concerns because we're not releasing it at particularly granular levels. So we've tried to be pragmatic because we're a little more restrictive. The privacy rules and the stronger, or should I say the share freedom of information rules are less broad in Europe than certainly EU than in USA, and so there was that concern. So I understand a number of marketing authorization holders have found the requirements and the requests to be difficult. There can be apparent conflict, but it's always important to note the source of the data. So whether EudraVigilance is your source or your destination should determine different actions. Was a kind of a message we try to get across. Over.
Thank you very much, Tom. Thank you for that input. I'm not sure where we are in terms of the Slido. The questions have been going on in the background. So the current question is, the ADR reports that I've seen from other organizations would suggest that they apply robust processes to ensure good quality ADR data. So again, there's, I suppose, mixed feedback here. I think the majority are neither agree nor disagree, so very neutral, and then reasonably split between agree and disagree. Now, we do have a bit of a way to go on this poll. We haven't touched on other topics. So we've touched on duplicates, we've touched on GDPR, but one of the key aspects that came around through the workshop was the integrity within each individual ICSR. So coding inconsistencies, narratives versus structured fields, gaps in information. So I would say it would be important to capture this topic in the chapter going forward. I don't know if any one of the participants or the speakers or panelists would like to discuss that topic, or I assume everyone would agree that the chapter would need to capture those topics. I'm not sure whether people are distracted by filling in the polls versus, I suppose, putting their hands up. Oh, our colleague from the UMC, Bjorka, if you'd like to come in on that one.
Yes, thank you. Yes, so inconsistent data within a report is a problem. For example, if a time to onset is reported as 3 months, but also you can calculate the time to onset from the drug start date to the reaction start date, and that one is 6 months, for example, then we don't know what the right time to onset is. And when you work at a location where you are in touch with the primary source, then you might ask that question, like, we have this conflicting information, can you inform us what we should keep? And that luxury, we don't have that as the end user. So if you could put this into the framework, and if that would help improve the quality, in the beginning of the process, that would help us in the end as well, because we have no way of knowing which information is correct in the end. Thank you.
Thank you very much. And Dave, I see that you have your hand up as well. So if you'd like to come in on this topic.
I'd love to. Thank you, and it's one where I'd offer a challenge for you in this chapter. One of the single most important fields is a field that we often receive cases with missing data, and it's a very easy field to complete. It's the Worldwide Case Unique Identifier, field C18 in ETBR3. Tom and I dealt with a case that was received from a third country, I'll be as generic as that, with no unique identifier, which became a mass of multiple hundreds, if not thousands, of clone copies and replicas. If the original sender had transmitted the primary key, field C18, none of those clones should have occurred. So I'd love for you to insist that every sender, every primary sender of a case adds a primary key in that field. And then just to take it down a notch, but to say pragmatic real-world evidence, we had a report from a patient just a few days ago, and we'd asked a question about congenital anomalies. Does your child have any congenital anomalies? And the parent had answered no. And literally in the very next free text field, she'd added that the child was due for speech therapy due to a cleft palate. So we've got to be very, very careful with questions and not validating patient information with a healthcare professional. So it's really levels of control that can be applied according to the data source. If I could make a plea for that.
Thank you very much for that input, Dave. Tom, I see that you have your hand up as well.
Yes, thanks. Two things there. Just looking at the Slido questions, I thought it quite interesting. Over 80% of the participants think their own organisations are good or very good with ADR data quality, but the subsequent question, about 80% were either neutral or disagreed that other organisations have good data quality, which I thought was quite a nice example of how people view things. Secondly, though, I had actually a question for Baukja, because I know for those who don't know, EEA data since 2017 has been sent via EMA to the WHO UMC. And one of the fields we don't provide is a narrative, we've mentioned that, but we do provide the structured data. And one thing you were mentioning, Baukja, is about inconsistent data. Where things are contradictory, just as Dave's example, lovely example there. And I was wondering if WHO have kind of database level checks, because you've got a similar database to us. And actually, I also know there's some colleagues from the FDA who are online as well, as other recipients of vast and diverse amounts of data, whether you have checks for these sorts of inconsistencies and contradictions within a case, because we're looking at developing such things ourselves, but we don't have it as a business rule. We're trying to develop them, and in the future it'd be quite nice to be able to have smarter business rules, but it's a sort of thing which is very difficult to proceed with, and I'd be very interested in hearing the views of particularly such organisations. Over.
Thank you. I don't think we have a process that would screen for inconsistencies, but sometimes we do need a rule to handle them. For example, with the time to onset, if the given time to onset is different from the calculated one, you need to decide as a rule, would you go with the one that is specifically reported or the one that you calculate yourself? And I would say normally we would adhere more strength to the reported ones than the data that we calculate, because that's more derived from the original report. But I don't think we screen for inconsistencies now. Thank you.
Thanks. I think we do the same in terms of if it's reported, we take that as the main thing, but we'll also have the calculated ones. And we've been discussing as part of we're updating our data analysis tools and considering what's the best approach, whether you have one or the other, or show both reported and calculated, potentially, giving people then the options.
Thank you very much, Tom. Thank you very much, everyone, for that discussion. I think we're at poll question number 9. So I think there's 2 more to go, but we are over time. I suppose just to sum up the discussion so far, I think we're all in agreement around duplicate check, GDPR, data quality within ICSRs, I suppose ensuring gaps, unique identifiers, etc. within ICSRs, key consideration around healthcare professional confirmation, data checks, etc. I haven't been able to keep an eye on the polls that have been necessarily going on in the background, but I assume that the feedback will be of use to the data quality chapter drafters. I haven't any other specific questions for topics for inclusion in the chapter at this point, but just before I close off the session, see if there's any specific points that other people want to raise at this point that we haven't covered in the session. And I would then just check. There's no hands up, so I'm assuming there's no burning topic that we haven't mentioned. We do. So is there one, I'm not sure, Anna, Lara, Tom, if there's another one question or one topic that you want to raise before I hand over to Paolo and others to close the session?
Nothing from me. Thank you very much. And thank you everyone for participating and for providing your feedback. It's been very interesting hearing your points of view and will really help. Thank you.
Okay, thank you. I will pass over now to Paolo for wrapping up. Thank you very much.
Thanks a lot, Anne. Thanks a lot to everybody. We would like really to thank everybody of you that participated today, especially meaning to all the chairs, the moderators, the speakers, the panelists that have made this event quite interesting and to have a very interesting discussion. As meaning we have heard a lot of topics today, a lot of different viewpoints related to any moment in the collection of EDR data, from the coding, from the managing, from the analyzing, for the follow-up. And we look at elements from SUSE, from simultaneous report to literature report as well, and also others. And meaning we will keep all this feedback that we received all together, and in order to draft the first version of the data quality framework chapter. And that will try to cover the full landscape of the ADR. And ideally, this chapter will be also helpful to standardize in a sense and to cover more aspects for each organization that is dealing with ICSR. As a next step, so we are going to draft the first version. we are going to review the existing practices, process, documents on data quality in the division domain, and that is something that we started already. Then we have organized this workshop, and therefore we got all stakeholder input. After that, around November 24, we are going to finalize the first draft. And we will start a public consultation at the beginning of 2025. And after that, after the public consultation, we will review all the comments received, and we will release the final version of this chapter. So with that, I would like to thanks again everybody, and we reach the end of the workshop. So many thanks. I don't know if Georgi or Tom or Anna have any point to add last minute, or anything from the panelists or the other chairs.
No, that was a really good discussion, and one that can continue. A very helpful insight. Thank you.
Thanks a lot, colleagues. We close here. Many thanks, and we will hear from us relatively soon. Thank you.